So good morning all.
What is the agenda? Is it someone going to speak some introduction first or before I start? Okay.
Yes, sir. Yes, Dr. Nalashiyam, students will take over. Good morning.
Okay. A warm welcome to each and every one of you to VIT's first virtual 8th National DNA Day Symposium. It is delightful to see so many of us coming together and being able to connect virtually during these unprecedented times. At this outset, on behalf of the Department of Biomedical Sciences, SBST VIT, I, Hridya Jacob, along with my counterpart, Aburi Prakirti, are honored to welcome our first keynote speaker for the day, Dr. Nalashivam Palanisamy. Thank you, sir, for being here with us today. I would also like to extend my welcome to Professor Bhaskar Rao KV, who is the session in charge for this occasion. Thank you, sir, for your presence. At this opportune moment, I would like to briefly familiarize you all with our keynote speaker. Dr. Nalashivam is currently working as an associate scientist in the Department of Urology, Vattikuti Urology Institute at the Henry Ford Health System, and also as an adjunct associate professor at the Michigan Center for Translational Pathology at the University of Michigan. Dr. Nalashivam completed his MPhil and PhD in human genetics from the University of Madras, Chennai, India, following which he successfully carried out his postdoctoral research in molecular biology as well as cancer cytogenetics in multiple institutes, including the University of Vermont, Burlington, the Miami Children's Hospital Research Institute, and the Memorial Sloan Kettering Cancer Center, New York in the United States. Over the years of his career, he has worked in the field of cancer genetics and molecular cytogenetics in various institutes around the world. His current research interests revolve around the discovery and characterization of gene fusions in cancer and their carcinogenesis using genomic technologies like FISH, CGH, spectral karyotyping, and NGS. His laboratory is currently focused on investigating the transcriptional and genomic architectures of solid cancer genome and incorporating the use of genome amplification to identify rare gene fusions formed at the boundaries of amplifications and deletions. He has also pioneered in the application of NGS technology for discovering new recurrent gene fusions with the primary goal of identifying novel gene fusions specific to certain cancer types. Recently, his laboratory discovered a new recurrent gene fusion in a subset of ETS negative prostate cancer as well. Apart from this, Dr. Nalashivam has also been an active member of the editorial and review boards of various esteemed and top-reviewed research journals. He has over 150 published research and review articles under his name. His contribution to the scientific community is immeasurable, and we are truly honored to have you among us today, sir. We're really looking forward to hearing your thoughts on the mysteries of the Human Genome Project. Before moving forward, just a humble request to all the participants to keep their mics muted throughout the course of the lecture, and I request you to post your questions in the chat box, which will be taken up by us at the end of Sir's lecture. I will now request Professor K.V. Bhaskar Rao, our session in charge for the occasion, to welcome our keynote speaker for the day with a few words. Over to you, sir.
Good morning to one and all present here. It is my pleasure to welcome all the participants in the first session of 2 days program of 8th National DNA Day Symposium. It is really a wonderful experience. On a weekend like this, Saturday, early morning before 9 o'clock, really 200 and above participants are joined. So that shows the enthusiasm of the participants to this program. And now I will welcome cordially Professor Nalashivam Palanisamy, Adjunct Research Professor from the Michigan Medical University, to deliver his first keynote address on mystery of the Human Genome Project. I welcome Dr. Nalashivam for your presentation.
Okay, can I share my screen now?
Yeah.
Yes, sir. Yes, sir.
Then I will share the video later. Okay. Okay.
Yeah.
All right. Okay, good morning, everyone. First, I would like to thank everyone, particularly Professor Radha Saraswati and the Dean and Chancellor, everyone, for giving me this opportunity. I'm really very delighted to talk to you all today. So this is going to be a kind of a general kind of talk. I'm not going to present any kind of high-end research data or experimental kind of findings so that you don't get scared away. So this is going to be a general overview about the human genome, and I will share my experience, actually dealing with this human genome ever since my graduate days from Germany. Because I learned to make chromosomes with the help of many eminent scientists like Dr. Chandra, I think she's in the audience. I started learning chromosomes, then I continue to actually work in the same field. So I'm really happy that I was able to make some terminal contributions to my research to various institutions, both in India and the US. So today, what I'm going to do is first, I'm going to give you a historical kind of perspective of the human genome. The reason I put the title as Mystery of the Genome is, as you know, that the genome, what we know so far about the human genome is just of the iceberg. As we get more in-depth technology or high-resolution technologies like next-gen sequencing and everything, I think we are getting more questions than answers about the human genome. So no technology is absolute, that is giving out all the information. Although we are able to complete the human genome sequence, that is only a tip of the iceberg. But there is so much going on after that, which I will kind of discuss this. Then I will touch upon how we are utilizing the knowledge that we gained through the Human Genome Project and other things, and how we are utilizing it to understanding the genetic basis of various diseases, both inherited and cancers. Then finally, I will close with some of the recent advances, and then I will conclude by showing some of the kind of future of the genome technology, where it will take us. Okay, so with that, I think, I want to, because we are celebrating this DNA Day. As you all know, there are so many kind of important breakthroughs in science over a period of time, ever since the humans started asking questions and making some research. There are innumerable discoveries, and I just listed very few, say like penicillin discovery or the electricity or the vaccines or the painkillers, all those things. I think we are not celebrating a day for those type of discoveries. They are just passing through, right? We just remember those. But I think the discovery of the DNA and the completion of the human genome is the one. I think there are many kind of research centers, they are celebrating the DNA Day. Why we are celebrating DNA Day? I think it is gaining more importance mainly because for any living organisms, rather I would say any living organism that is moving around in the world, whether it is like humans everywhere or the plants, they are standing in one place, but they are moving vertically and also horizontally. For any moving living organisms, I think DNA is the fundamental molecule. Without DNA, no living organism can exist. Only with the good combination of DNA, there are remarkable individuals who are really gifted and they are making these important discoveries and all those things, although every individual is unique in terms of their DNA structure and everything. So given the fundamental nature of the DNA as the unit of all the living organisms, I think it is appropriate. It seems like as we are living, I think we are celebrating the DNA every day rather than just on one day, because it is the DNA, what we have is the one that is making every individual who they are, like that. So with that importance, I think, let me move on. Before we celebrate everything, I think we should not forget the people who really laid the foundation of all these discoveries. I think I always recognize in my lectures, I used to kind of mention Gregor Mendel, who was the father of all this modern genetics, who introduced the concept of the inheritance with the law of segregation, law of independence, law of assortment, and everything. He laid the foundation stone, and he was the one who identified the existence of the gene or the factors that is responsible for carrying on the characters from one generation to the next generation. So it took a while. And then during that time in 1888, Vellore, he recognized the presence of chromosomes in the cells as dark bodies. Even at that time, they didn't know that the DNA or the genetic material exists within the chromosomes, but they were able to recognize those things. But it was, I think the breakthrough came in 1944. I think all the biomedical students who are the first year or second year, I'm sure you're all learning these experiments in your class. So these three people, they made this remarkable breakthrough discovery in 1944, showing that DNA is the genetic material by using this bacteria as a model organisms, and they showed that the DNA is the genetic material. So I don't have to go through the details of the experiments. As students, you're all learning this. So even until now, we don't know the structure of the DNA and other things. Then the breakthrough came with this picture, Dr. Rosalind Franklin, who was technically an expert in making this excellent X-ray diffraction and all these crystallographic pictures. But she had the idea that the DNA is a helical structure. That's all she had it, but she did not really solve the problem. But it was in 1950s, James Watson and then Francis Crick, they are the one, they got the picture from Rosalind Franklin's office. I think you should read the book Double Helix, written by James Watson, how they got the picture and everything, So there is a big story behind that. But anyway, when they got that picture, they were able to give the interpretation of the DNA double helix. Because before that, there were kind of models that were proposed that the DNA could be triple helix or this and that, but none of those were not, accepted by the community. But it was actually Watson and Crick who were able to give the kind of I think the proposed structure. I think the main reason for their structure, because they feel confident that this should be the DNA double helix model should be the right model, because they didn't show any experimental proof saying that this is the right model, but at that time they just made the proposal that this could be the right structure. Because the reason they gave that this should be the right model is that when you read this paper carefully, there is a small paragraph which I highlighted in the red box, which shows that it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. I think that is the key, because the DNA making its own copy, that's where the life, that moves on from one generation to the next generation, because every cell, when it divides, it has to make its own copy of the DNA before they go to the next cell division. I think this is the remarkable kind of observation that they are able to make, and which made it, which was later proved that this is the kind of the right DNA structure. It was really amazing. So when I read this article, I encourage all the students, please, if you have not read this article before, please download this. It is available on the internet. Please read carefully. This is really fascinating. So I was really intrigued. How a critical kind of observation, that you can make, because I think before that, many people have looked at this X-ray crystallographic picture and everything. They are not able to come up with the right model. but it was Watson and the Crick. They were able to make this remarkable discovery. So I was actually fortunate to meet with Professor Watson in one of the meeting in Washington a couple of years ago, where I was giving a presentation. He was sitting in the audience, and after my presentation, he came and he pat me on the back, and I was fortunate to spend at least an hour with him talking about all these things. It was really a dream come true moment for me. It was remarkable. If anyone comes to the US, I think he is still living in New York. I hope you will get a chance to meet with him again. So since then, from 1953, within another 50 years in 2003, they initiated the Human Genome Project, and the entire human genome sequence was completed. Although initially this project was started by James Watson, and with some conflict of interest or whatever it is, he left the project. Then Dr. Francis Collins, he took over, and they were able to complete the first draft of the human genome sequence. So I was again fortunate to meet with Francis Collins when I was in Singapore, when he was actually presenting his work at the time in there. It was in 2005, I met with him there. These are all very important moments in my life, being a geneticist working inside the genetics field. Then I think we are celebrating this 20th year of the first draft publication of the human genome, the complete genome, which was jointly released by Francis Collins and Craig Venter at the White House. So I think if you get a chance, please read the February 5th issue of the Science magazine. So they have a lot of nice, kind of write-up about this project. I think it will be very useful for the students, and there's a lot of interesting observations that are published in that magazine. So we have the sequence now. We have the complete human sequence. So I would like to highlight upon what are the different sequence components that is making up the human genome. Okay. So as you all know that humans being a highly evolved organisms, before the Human Genome Project, people thought humans may have hundreds and thousands of genes, which makes up the human genome. But interestingly, when the Human Genome Project was completed, then now we realize that there are only about 20,000 genes, that is protein coding genes, that make up the human genome, which accounts for only about 1.5% of the 3 billion base pair. Okay, so therefore, anywhere from 98 to 99% of our entire genome, it is there for a reason, right? What is it doing? So they must be doing something, but now with the advent of the next-gen sequencing technology, we were able to show that there is so many non-coding RNAs that are lying within this, the other 98 or 99%. So now they realize that initially people thought only 1 to 1.5% of the genome is transcriptionally active, but now they realize that more than 99% of the human genome is transcriptionally active, with the discovery of so many these non-coding RNAs particularly. I think that's where all those sequences are there for a reason. So when you look at this non-coding RNA landscape, there are thousands and thousands of long non-coding RNAs, small RNAs, microRNAs. Here I listed all of them with the numbers. There are, because the rest of these, other than this 20,000 protein coding genes, the rest of them are occupied by these small non-coding RNAs. Then now I think people thought we really don't know the exact number of the non-coding RNAs that are present in the human genome, because these non-coding RNAs are not, and all the non-coding RNAs are not expressed in one type of tissue. They are coming from different type of tissues. There may be different kind of non-coding RNAs may be expressed. And so as the term non-coding RNA means, these molecules are not really coding for any proteins. I think that was initially, people thought that these non-coding RNAs are strictly, they are not coding for any proteins. But recently there are evidence clearly show that there are many non-coding RNAs. They are having small, because these non-coding RNAs are small molecules. They are less than about 300 or 400 base pairs. So the proteins that are produced from these non-coding RNAs are very small. So now they call it as micropeptides. That is the new kind of concept that is emerging because of the sequencing, and they have changed the new algorithms to find out the open reading frame in the RNA. So there are new micropeptides are being discovered. So we don't know the exact number. Definitely they are in, at least so far we know about close to 40 to 50,000 of non-coding RNAs in the human genome. But I think the numbers are not final. I think the more work is going on. So as you know, the type of non-coding RNAs, there are different names, like you have siRNAs, pRNAs, microRNAs, all those RNAs are there. And I think since we don't know the exact number, because it differs from different tissues, and also it may be differing from normal genome to the human and the cancer genome. So I think a lot of work is being done. So far, whatever that is published in the literature is more anecdotal evidence of characterizing the function of non-coding RNA, but the number is not finalized yet. And most of the function of the non-coding RNAs have not been validated yet. So now it is very clear that based on the initial evidence that it is clear that there are many publications clearly show that these non-coding RNAs are thought to contribute to diseases, including cancer and Alzheimer's disease as well. Okay, so coming to some of the functions of this non-coding RNA, many non-coding RNAs show abnormal expression pattern in cancer, meaning that these non-coding RNAs are not expressed in the normal tissue, but you will see them expressed only in the cancer. And some of the I think functions of non-coding RNA. I think one of the non-coding RNA that I was involved is the NEAT1, 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 that is an important non-coding RNA. We clearly show the function of this non-coding RNA, important role in breast cancer. And the main function of this non-coding RNA is by regulating some of the protein coding genes. Because I don't have time to go through the details how exactly they are regulating the protein coding genes, but I'm sure you guys will learn all those things in your class. And then so the perception that these non-coding RNAs are junk is no longer valid because they are truly a functional transcriptional products. With important some regulatory role in the cells. I think in cancer, there are many non-coding RNAs are identified as oncogenic drivers, and some of them are tumor suppressors in every major cancer type. So I think that is the main focus, because we have investigated extensively almost all the 20,000 protein coding genes. We know the complete structure, the intron exon structure, the protein size, the protein structure, most of them. So I think now the current focus of research is understanding this non-coding RNA. So there is a lot of laboratories working on that. So the other important kind of type of RNA in our human genome is called the pseudogenes. I don't know how many of you are really familiar with this term, pseudogenes. This is another class of non-coding RNA, which was completely ignored for a long time, which people thought these are all just a junk portion of the DNA, but it is not so. So now we know that there are tissue-specific and cancer-specific expression of pseudogenes, and both in normal tissue and in cancer as well. So originally, the pseudogene was first identified in 1977 from this frog, Xenopus levis, by Jack et al. In 1977. So basically, the pseudogenes are not typical non-coding RNA, but essentially the pseudogenes are basically some dysfunctional relatives of known genes, meaning that there are wild-type genes like protein coding genes, which is there, but during the evolution, because the human genome is a dynamic molecule, it is constantly changing, there are new genes are produced over a period of time. So, and some genes, they may lose functions like that. So So far, based on the sequence data, there are 3 different types of pseudogenes are recognized in the human genome, which accounts for about, right now, there are about 20,000 genes. I think the numbers are more than 20,000. So there are 3 types of pseudogenes, of which one is called the process pseudogenes. I will just explain the essence of time. I may not go through the details, but I will tell you how the process pseudogenes are produced. So basically what happens when a DNA molecule is transcribed of a particular gene, the complete messenger RNA is produced, right? With all the introns and exons. The first initial transcript is made, then it goes to the splicing. That means removing all the introns and all the exons get kind of joined, and it makes a functional messenger RNA. Then it moves to the cytoplasm and it is making protein. So what happens in the cell immediately after the transcription, then after the kind of splicing and everything, when all the exons are staged, what happens in the cell for unknown mechanism, we don't know the exact reason yet, the single messenger RNA molecule, it gets reverse transcribed. So meaning that the single messenger RNA is converted into a double-stranded DNA with the full length. So now all the exons are removed. All the introns are removed. Now all the exons are fused from the single messenger RNA converted into a double-stranded DNA now. So now the double-stranded DNA, what happens, it gets inserted into a different location in the genome. So essentially, a functional gene in one location of the genome. So now after making kind of a reverse transcription, after making a new DNA double-stranded molecule without the introns, it gets inserted into a different location in the genome. So this is called processed pseudogenes. Process means all the introns are removed, so the exons are fused and the double-stranded DNA made, and it gets inserted into a new location. So like that, there are many genes in our human genome. That some of the genes have at least 15 pseudogenes located in different locations in the genome. So we thought that these pseudogenes are just kind of error in the transcription like that. But now we clearly know that because of the function that these pseudogenes do have a function, some regulatory function. They are actually expressed in a tissue and a cancer-specific manner. Okay, and there are other class of pseudogenes, so like non-processed pseudogenes. These pseudogenes are formed just the duplication of the segment of the DNA in one region of the human genome, which contains the full coding sequence of a protein coding gene, which is duplicated and get inserted into a different location. So this is called non-processed. So that means it retains all the introns and exons. It moved into a different location. This is called non-processed pseudogenes. And the disabled pseudogenes are the ones that these genes are not transcribed or duplicated, but in its location itself, they get disabled, meaning that some of the promoter sequences are undergoing mutations, which make it incapable of actually activating transcription or something like that. So these are all some of the genes called disabled pseudogenes. This is entirely a new class of genes which are recognized in the human genome, which was not identified for a long time. So this is the new discovery that was made because after the kind of next generation sequencing, because I think before that there were many anecdotal evidence, like they discovered in the frog and some even in the human, there are few pseudogenes were recognized. But now we have I think we were the first group from the University of Michigan. We showed the complete whole genome level expression of pseudogenes across several, at least 13 different cancer types. When we did the next gen sequencing analysis, we published that paper itself. Then recently, I was in my lab, we actually discovered a new gene fusion that is particularly expressed only in prostate cancer. This is called the KLK4, KLKP1 pseudogene that is found only in prostate cancer, not in any other cancer, not in any other normal tissue. So this is an important biomarker that we discovered in my lab, which we published last year, I think in 2019. So anyone is interested, please read this paper. Because this is the first evidence that we showed that there is a pseudogene, which we thought that is non-functional or junk DNA. Now we clearly show that the association with the cancer. Okay, so this is the first evidence in the history of the analysis of the human genome that we reported for the first time. Okay, so now, so let us go into the little details. So this, I think this slide shows the more details. So as I told you, when we complete the human genome sequence, about 3 billion base pairs, we know that there is only 20,000 genes. Okay, so this is only just knowing the sequence of the entire genome of any organism, not only humans or any organism. That is only a tip of the iceberg, because there is so much, because the DNA molecule is an information-packed molecule. There's so much information is actually kind of packed in the DNA molecule. But when the information is released, and there is so much going on. So what happens, because we thought that humans being a highly complex and highly evolved organism, we thought there is more than 100,000 genes, but it is not the case. As I told you, it's only 20,000 genes. So how this complexity is actually carried out in actually just maintaining an organism from fertilization to a fully functional organism, okay? So the information is gradually released by the transcription. Essentially what happens is one gene, so for example, here it has like 4 exons, it is getting transcribed. But from a single gene, more than one type of transcript can be produced by a mechanism called alternate splicing, or what we call actually splice variants. Like one mRNA transcript, it may contain exon 1, 2, 3, 4. Then other transcript may have exon 1, 3, 4. Other transcript may have exon 1, 2, and 4, like that. So from a single mRNA, from the pre-mRNA, multiple transcripts can be produced. So with that accounts for about, currently they estimate about 70,000 proteins. From 20,000 genes, there are about 70,000 proteins can be produced because of all the splice variants and everything. Then when the protein are produced, they further undergo several post-translational modifications like like phosphorylation, acetylation, and all those kind of post-translation mechanisms. That adds more complexity to the proteins. Then it accounts for about almost about 1 million proteins. Okay, so this is how the enormous complexity of the human genome is kind of exhibited, and that adds to the complexity of the organisms. I think this is the big mystery, that we don't have a complete understanding of how many of these splice variants are expressed in each tissue. So right now we have information only about how many genes out of these 20 genes, how many genes are overexpressed in a particular tissue and how many genes are not expressed in a particular tissue. But we don't have a complete understanding about how many kind of splice variants are produced from each gene, and how these splice variants are restricted in kind of tissue level or cancer specific expression, all those things. There is so much work need to be done here. We don't have a complete understanding about this. So this is called, when a protein undergoes all these, what is called the post-translational modification, they are called as proteoforms. So there are 1 million kind of proteoforms, and they are functioning to keep us going. So here is one example to show how this is working. So in this cartoon, I show from one gene, then there are more than one RNAs produced, like mRNA1 and mRNA2 with the different exons. So they are called isoforms, or actually splice variants. And each one have entirely different sequence. Some of them have a single nucleotide polymorph, some also variations, and some they undergo glycosylation. There is a post-translational modification or phosphorylation, and there are different type of proteins are produced from a single gene. So this is how from a single gene, multiple proteins can be produced to exert complex functions. Here I'm quickly show you how what is called the post-translation modification is happening with the insulin gene. So first, the ribosome basically translates the mRNA sequence into a protein. It's called the proinsulin. Then it passes through the endoplasmic reticulum, where it is get cut and folded and held by disulfide bonds. Then the protein passes through the Golgi apparatus, packed into a vesicle. Then the vesicle more parts are cut off and it turns into a mature kind of insulin. It goes through all those modifications before functioning as that. So like that, every protein has to go through this process with different functions. So this shows the clear complexity, how the information from the DNA is actually flowing through the several mechanisms to exert different functions. So now we know that the human genome is sequenced and all the information, we know that how the DNA information is actually translated into protein and functioning. So now I think this will be useful information for the students. The sequences, as I told you before, although we have 20,000 genes, all the 20,000 genes are not expressed in all the tissues, right? The genes have some tissue specificity. Some genes are expressed only in one particular tissue and other genes are not expressed at all. Like that, in order to answer that question, to see what is the function of all the 20,000 genes, the NIH, they invested money to sequence the mRNA from 53 different types of human tissues, including heart, liver, intestine, and all those different organs. They selected different organs and they sequenced 53 different type of normal tissue, and they have completed the sequence of all these tissues, and the entire sequence is available through this portal. This is available freely. Anyone can download. You don't have download. It is already processed. You can just view the data, just by click of the mouse. I will show you the next slide. So this is called the Genotype Tissue Expression, or the GTEx portal. So I think the students, you can do a lot of projects. If you have some little bioinformatic knowledge, definitely you can develop a lot of projects looking at this database. Then similarly, there are about 25 most common cancer types, and approximately about 500 samples for each cancer type have been completely sequenced through this what is called the Cancer Genome Atlas project. And they again funded by the US government. It was completely sequenced, and all the data is freely available. This is called the Cancer Genome Atlas project. It is available through this NIH portal. You can visualize this data. And all this data, you don't have to be a bioinformatician. You don't have to download the raw data, because all these data are highly curated, and you can just view the results. I will show you here. So anyway, I'm sure you are familiar with this database. It was called the UCS Genome Browser. Anyone can go and look at the exact physical location of all the genes for any chromosome, any particular region. What are the genes that are located? So the top, the horizontal line is the chromosome, and the box that is highlighted in red is the small region that I focused. And below that, all the genes located with that region is highlighted here. And when you look at all at the bottom, very bottom, there are like black lines here, which shows the sequence from multiple organisms. So they are trying to understand the conservation of the DNA across several species. As you can see from the very bottom, there are actually few kind of black dots here, and there are many white spaces here. So what I'm showing here is, when you see the black line from the top to bottom, everything is like, that means the exons, the region that is coding for a protein, is highly conserved from the lower organism to the higher organism. But the intronic sequences are not there in the lower organisms, and those are all added starting from the elephant, dog, and the mouse, and a rhesus monkey, like that. So as I told you, the genome is a highly complex and dynamic. It is constantly evolving, and new genes are added and some genes are eliminated, like dysfunctional units like that. So this clearly shows the kind of conservation of the gene structure across several species. So you can actually just mine through this database. If at your time, there are so many links at the bottom. of this page, and you can visualize it. If anyone needs help, please contact me. I can give you a tutorial how to mine this data. So next, this is I'm showing just one example. When you look at the GTEx portal, as I told you before, you don't have to download the raw data. All the data is processed and analyzed. It is ready for visualization. If you go to this portal, you can just type the name of the gene that you're interested. Just with a few clicks of the mouse button, you will be able to see what is the results. So here I'm showing an example. This is a pseudo gene called the KLKP1, which shows the expression only in normal prostate, and it is not expressed in any of the other 52 normal tissues. This clearly shows the tissue-specific expression of a gene. So there are several genes like that. They are expressed only in a particular tissue, but you don't see them in many. And there are multiple genes. They will express across all the 53 tissues like that. So I think any students who are interested, if you focus a particular organ, if you want to characterize what are the important genes that are upregulated or downregulated and try to understand the function, you can validate those data in your laboratory experiments. So this is a nice kind of resource for the students to mine. And I think when you have free time, instead of going through the internet, clicking some unwanted sites, please spend some time just going through this portal, because these are all just few. There are several links. You just click, and when you go through several times, and you will get a good sense of what this database means. And I'm sure you will be able to kind of develop some interesting projects out of this. And similarly, this is the database called the CBio portal. All the cancer data, the cancer genome atlas data, is all the data is curated. You're not looking at the raw data. Everything is already analyzed, processed, and normalized, and everything. There are data from 89 different cancer types are available in this database. Suppose if you want to see a particular gene, what is happening across these 89 cancers, whether it is RNA level, whether it is overexpressed or it is mutated, what is the copy number at the DNA, everything you can look at it just with a few clicks of the mouse button. You will get the nice kind of box plot. So here I'm showing a result of a PIK3CA gene, the mutation. As you see, across these 89 cancer type, only the first 10 or 12 cancer type are showing high level mutation. I think the green bars are showing the mutation, and the red is showing the amplification of the copy number. This is at the DNA level. So you will get the data right away. You don't have to kind of go anywhere, do any experiments or anything like that. All the data is readily available, and you can actually try to develop some projects. So once you get some of these, and if you have a good collection of tumor samples from different population, like from Vellore population or Chennai population or anything, you can validate these findings because there is some lot of kind of population or racial kind of differences in the incidence of all these mutations from other markers. So you can validate this data in your lab. I think this will be a nice project for the MSc students and other things. You can just use this database for that. I'm sure you're already familiar with that. If this information is actually redundant for any of the students, please forgive me, but I think it is my duty to show what is going on. So now we know what is the nature of the genome, how many genes are there, how the information is flowing, and we sequenced the entire DNA and RNA and everything. So now we are trying to understand what is happening, what we can do with this information in terms of understanding the role of these genes in different genetic diseases. So please go to this database again, Online Mendelian Inheritance in Man. Here, this database contains information of all the genetic diseases, how the disease was described initially, what are the genetic mutations that are underlying this, then all the corresponding literature that is available so far that is done on each diseases. You will get all those information from this database. This is a very important resource for anyone to look at it. So now this is the important point. So now given all this information that we have gathered from the Human Genome Project and other things, the US government, they have started called the Genetic Testing Registry. So they are maintaining how many tests are being actually performed for different diseases, what are the different genetic conditions, how many genes are involved, and how many labs are involved. So I just updated the data as of last week. There are about 77,000 tests have been performed for about 17,000 different genetic conditions. and which involves about 18,000 genes. Almost every gene has something to do with different diseases. And there are about 550 labs that are well approved and well regulated labs in the US. They are actually conducting this test. Okay, that clearly tells the enormous complexity of this operation, and it's very useful information. It is constantly updated. I think every weekly you will get the most updated information from this database. All right, so I'll quickly go through how all different type of genetic testing and everything is done using this information. So as I told you before, for every organism, the DNA is the fundamental unit for any moving organisms. So any ailment or any disease or any conditions, it has to have some genetic basis. Okay, I think that was actually Professor Marimuthu when we were students. He always says that even road accident is a kind of, there is some kind of genetic basis. Only some person is mad or something like that. It has something to do with the genes. That's why he's kind of causing accident like that. So everything has some basis in our genes, every ailment. So it could be an inherited condition or acquired medical condition like cancer, or inherited means some genetic diseases are kind of inherited. So it again caused by a change or mutation in the individual DNA. So I think with the completion of the Human Genome Project and several other research, that really helped to understand the multiple factors that are acting together. It is not only genetic, sometimes both the genetic and environmental component is involved in the causing disease, like complex disease like cancer, diabetes, and heart disease and everything, right? So, so now all the research that is focused on the genome that is really enabled for the development of improved diagnostics and kind of therapeutic strategies. So I'm just jumping that, in between there are so many things happen, but recently, I think the current status of the way that we are doing genetic testing and treatment and other things is what is called personalized medicine or the precision medicine. Although we all have the same kind of DNA and everything, but we are not the same. Okay, so every individual has some kind of variations in the DNA, which make them more susceptible or kind of resistance for each disease. For example, like if people have a kind of a single nucleotide polymorphism in the gene called CCR5, and they never get infected with the AIDS virus. They give natural protection just because of the polymorphism, and some individual, they may not have that polymorphism. Like that, every individual is unique. So therefore, it is important to get our sequence information, complete information, to decide on what type of treatment that each individual will be eligible for that. So this is the emerging approach in most of the US and the Western labs are being doing. I'm sure in all over the world, I think this is a common practice that you have to investigate the biopsy material for any genetic changes. Then based on the treatments are decided. So this is really helpful to predict more accurately which treatment and prevention strategies for a particular disease will work in which group of people. So I think this is the kind of emerging approach. This all again based on the changes in the DNA. So this is what, so you have to do kind of multiple testing, whether you can do an next-gen sequencing or PCR or FISH or immunohistochemistry or whatever the type of test that is required to test the genetic changes that is involved for each individual. Because this, we are going to do the test based on the markers or the biomarker or the gene defect that we know that could be associated with a particular disease. So you have to identify each individual's which marker they are positive or negative for that. If they are positive for a particular marker for which there is a drug available, that patient will be directed to that particular treatment. And if the other patients, if they are kind of negative, there is no drug available. So even if you give the already available drug, it is not going to work like that. I think this is really helpful. That's where the DNA screening is very important for doing the personalized or the individualized medicine. So when we do this kind of screening, it is not always DNA. So you have to remember that all the defects are not lying at the DNA level. So it could be just overexpression of a particular RNA. It could be a particular mutant kind of RNA. or it could be some non-coding RNA, like we said, that is responsible for cancer and other things. So we are not looking at just the DNA alone. I think that is the main reason that every disease condition is extensively investigated at the DNA, and they have completed the sequencing of RNA and everything, so that they can identify whether there is a DNA amplification or a deletion or translocation, or the DNA that is a mutant RNA is there, or the RNA is overexpressed. Or sometimes the DNA and RNA may be perfect, but the protein, because of the post-translational modification, other things, it could be abnormal. So sometimes at the protein level, we have to make the diagnosis. And recently, importantly, there is a new field that is developing. It's called metabolomics. There are many intermediate metabolites from different cells, like from the metabolic pathways. What we found was there are certain metabolites get accumulated because of the defect in the pathway. There's a particular enzyme. If the chain is kind of blocked, then any metabolites before that will get accumulated, and that will become responsible for a particular type of disease. So all, all the metabolomic screening is a new field that is highly kind of developed by using the mass spectrophotometry technology. There are a lot of laboratories are doing the whole genome-wide, screening of metabolites from the blood samples, from the tissues and other things. And there are many important discoveries have been made at this level. So it all depends on which marker is affected in a particular disease, and you can do the appropriate test to identify the actual, kind of defect, what is going on there. I think somebody is raising a hand. Is there anything I need to be cautious about that? Radha?
Sir, if they have a question, they can post it in the chat box. We will get back to them at the end.
Yeah, I see the Harihara Sudan. He just raised a hand. I don't know what is for it.
Maybe by accident. Yeah, at the end only we will take the questions, sir. You can continue.
Okay, no problem. Okay.
Yeah.
All right. All right. So now, what is a kind of genetic testing? Basically it identifies the changes in chromosomes, genes, or proteins. So the results of a genetic test will conform to rule out a specific kind of genetic condition and to determine the person's chance of developing or passing the genetic disease like that. I think that's where the genetic counselors and all those people are coming into play. Let me see, I think this is blocking me. Okay, so I think I don't have to go through all the details about the genetic testing. I'm sure you're all familiar with all those things. I think this is blocking. Okay, so let me skip this with the sense of time. I may not be able to go through this. Yeah. So how the genetic tests are done, I think this is more important. This is not like doing a blood test for blood glucose or alkaline phosphatase, this and that. I think genetic test has to be done very carefully. It has to be kind of recommended or kind of prescribed by a doctor who suspect what type of genetic testing is appropriate. I think all this testing is very much regulated in the US and the Western world. I don't know how well it is done in India, but here it is, everything is strictly controlled. The doctor, the primary doctor, he orders the test, and first the genetic counselor is go through the family. They collect the family history, everything to understand the genetic risk. What are the possible inheritance to the next kind of generation, everything. So I think depending on the type of test, they can either get blood sample or the skin biopsy or amniotic fluid or whatever the samples that is needed. And either they can do the test on the chromosome or RNA or the protein, depending on the suspected disorder. And also the newborn screening, which is quite popular in the US, mostly done on small blood samples. Most of the time they're taking the heel, pricking of heels. And also in order to assess the kind of status of the carrier status, most of the if it is inherited conditions, most of the time, both parents will be kind of tested as well. So there is a kind of kind of procedure to do that. Even before doing the test, they have to get the informed consent from both the parents and the patients. everything is regulated in the US. So the way the genetic testing was done before the DNA information was completely understood in the early kind of 50s and in the most of the time, I think our knowledge is limited only to the chromosome. We know the exact number of chromosomes in the 1950s and 60s. So I think most of the genetic testing is restricted only to the chromosome level. And today there are more than, I think I cannot go through the details. There are so many kind of specialized genetic testing going on, okay? It could be just for diagnosis or predictive or presumptive genetic testing, carrier testing, prenatal testing, even pre-implantation genetic testing is more common now, you know. If they have any kind of an affected genetic defect child in the previous pregnancy, I think they can do some pre-implantation. They can do the individual cells. They can then they can implant this embryo back into the uterus to develop the child like that. There is a well-developed procedure for doing that. And newborn screening and pharmacogenetic testing. All those are really the kind of recent developments because of our knowledge of the DNA. I think what is the future? I think in the future, genetic testing will be an important part. Since we have a good understanding about most of the genetic disease, more than about 7,000 genetic diseases are there, so we have a good understanding about what are the genetic defects. So the genetic testing will be important before they do anything with that. I think for that reason, I think the genetic test registry that is developed at the US is a very valuable resource that will provide a centralized location for test developers and manufacturers to submit information on the test. And the patients and the healthcare providers, research and others will be able to search the registry for information or indications for use, clinical validity and utility. This is a very powerful database. You know, I think students, when you have time, please go through this. You know, you will get a lot of valuable information. And then the cost, I think that is the most important factor. I think at least in the US, they have a good kind of insurance coverage. But in the developing countries, I think if people have to bear the cost of out-of-pocket expense, it may be expensive, but since the sequencing and other thing are kind of making it more affordable, I think the genetic testing will be more important, very useful in the future. I think that's the reason that we are living in a time that is more effective of personalized medicine. So we don't have to. We cannot generalize, like one person has this disease, oh, okay, he had the same kind of disease. You cannot talk that anymore. Every individual is different. You have to get tested on a personalized basis. There are about 7,000 genetic conditions have been recognized, and more than about 500 labs that are doing in the US all this testing. I think there is a registry for that we discussed before. Okay, please go through that. So the critical factors that are important for the clinical diagnosis is important, okay? So now we know that each genetic disease is identified with a unique set of molecular alterations. So the current approach, like we discussed, is a stratified medicine or the personalized medicine. And for many conditions, there are targeted therapy available. It may not be available for every conditions, but for several of those, there are targeted therapies available. For that reason, so you have to detect any abnormality with accuracy, you know, whether it's a mutation or a DNA copy number or gene fusion, you have to do, the test should be high quality and it should be available conveniently for everyone. And the test should be performed at a fast and accurate manner. And any lab that is doing, you know, you have to trust and you have to have a confidence about the lab who's doing it. You cannot make any room for any mistakes. So I think it is understandable and useful information they have to present so that anyone can, both the doctors and the patient can understand. So these are the common practice that are very rigidly followed in the US, and it is being routinely done here. So we have come so far about the genome. We have the good understanding about what is going on in the normal cells and other things. So now I'll talk a little bit about the cancer. As we know, cancer is called the disease of the genome. Okay, it has to happen something with the genome. Because the genome can't be normal, then you get a cancer. Every cancer sample, either there should be a single kind of genetic abnormality like a small gene mutation or a one chromosome translocation or multiple complex operations. There is no single cancer cell with a normal chromosome. It has to have some genetic mutation. So basically these cancer cells, they completely lose their control. The normal cells, they go to individual checkpoints for cell division checkpoints. They lose all those controls. They just grow uncontrollably. So I think that is the fundamental question that we don't have an answer, how a normal cell is transformed into a cancer cell. What is the earliest or the initiating event that make a normal cell into a cancer cell? That is the big fundamental question which nobody has an answer. Because the reason is most of the cancer, when they are at the early stage, they are asymptomatic. We have no idea. They don't show any symptoms, no fever or weight loss or this and that. We don't know the cancer is actually growing in our body. Only the cancer grows to a critical stage. Okay, when it started affecting other functions in the body, then you started getting some weight loss or continuous fever, this and that, or accidentally when you do this master health checkup or some scanning, you accidentally found out some mass or something like that. When you take a biopsy, then you see that there is a cancer like that. So when you recognize the cancer as a symptomatic disease, which is too late, which already actually grown into a level that which is very difficult to control. So as long as the cancer, whatever the cancer that is identified, which they remain in their native location, it may be easy to control because you can either surgically, you can remove that particular tumor or you can give a focal radiation therapy or something like that. But once the cancer cell started moving from its primary location to a different kind of parts of the body called the metastasis, that's where it is difficult to control. Okay, so this is the important question for the, I think the new generation of the scientists who are listening. You know, this is the biggest challenge. We have to find out what is the earliest event that really transform a normal cell into a cancer cell. Okay, so I think that is, there are so many models people have tried, but still we don't have a good answer. All we are dealing with, all the cancer is the cancer that has already crossed the threshold and they reached a limit where we have only kind of a little control in their growth. So I will just walk you through what are the challenges that we are facing in the cancer field. As you know, like I said, cancer is the disease of the genome. Okay, it has to have some genetic abnormality at the DNA level. It could be at the structural chromosome, like because the chromosome number is not strictly 46, it could be 48 or 49 or 60 or 100, whatever it is. There are so many deletions or a particular region may be amplified like that. So both the numerical and the structural changes are fundamentally kind of essential in any cancer cell. So this is the first chromosome abnormality that was ever recognized in cancer. So this is a simple chromosome translocation that was identified in the 1960. First, it was observed by Peter Noble and David Hungerford in Philadelphia. They identified from the patients with what is a disease called chronic myeloid leukemia. It is a kind of blood cancer, where they identified a small chromosome That time, we don't know because in actually 1960, there was no chromosome number. They were able to count the 46 chromosome. They consistently found there is a one small chromosome, which is always abnormal in all the patients with the chronic myeloid leukemia. This was the observation in 1960. And then in 1973 or something like that, Dr. Janet Rowley from University of Chicago, with the advent of the banding technique, she recognized that the deleted chromosome is not really a deleted chromosome. The portion that is missing from this deleted chromosome actually translocated to a chromosome 9. So later with the banding technique, they recognize that this is chromosome number 22, the small chromosome. The portion that is missing here is actually moved to the chromosome 29. So this is called balanced reciprocal translocation. So as you can see, all other chromosomes are perfectly normal except this one chromosome, chromosome 9 and 22. This is a simple translocation, what is called balanced translocation. Here, meaning that there is no loss of genetic material. Only thing, they are actually moved to a different places. So essentially, the chromosome has to break at the same time on each chromosome, both 9 and 22. Then the parts are exchanged perfectly. So this is the only abnormality that you will see in 100% of the patients with the chronic myeloid leukemia, with this disease. This is the only genetic abnormality that is causing this disease. So what is happening in this translocation? So when there is a chromosome break point, at the break point, there is a gene affected, okay? And the chromosome 9, there is a gene called ABL, and the chromosome 22, the BCR. So the entire normal gene, the ABL, is from chromosome 9 is translocated to chromosome 22, and it is placed under the control of the BCR gene. So now the gene is under the control of a new promoter, which drives the overexpression of the ABL gene. So the abnormal level of the normal ABL protein is responsible for chronic myeloid leukemia. As simple as that. So when the gene, the ABL gene, if it is in the normal location, it is doing its normal function. Okay, just because it is going to, it is like a bad company, right? You know, when you are going with a bad person, you know, you get influenced with that and you do all the bad activities. That is exactly what is happening, you know. So this is the main event that causes the blood cancer. So this is the remarkable story. Starting from the chromosome discovery, now they have a drug. There are so much actually went through in the research because they did several molecular cloning. They identified exactly the BCR-ABL, what is the fusion. I think in the from the 1960, I think in the 1990 itself, there is a drug that was developed because the ABL gene is a kinase, is an ABL tyrosine kinase. So like you see, when you have a normal protein, you have a receptor and it gets phosphorylated. So now they have a drug that goes and binds to exactly in the pocket that prevents the phosphorylation of this protein. So basically using this drug, you are inactivating the function of this ABL tyrosine kinase. By that, you can actually treat the disease. You are not curing the cancer because the fusion gene, you cannot remove the fusion gene, but you can prevent the function of the abnormal level of the ABL tyrosine kinase. So this is a remarkable, kind of work. Even the first patient who received the drug, I think about 17 years ago, when she got the drug, that patient is still alive. So every day what she is doing is she is taking a pill of like 400 milligram, like you are taking a blood pressure medication for one pill like that. As long as actually she continue to take the medication, you can suppress the abnormal level of this protein, she get actually, she's living cancer-free like that. This is a remarkable discovery from the identification of chromosomal aberration to the drug development, okay? So this is the simple story because in the CML, there is only one chromosome abnormality or one chromosome translocation that is responsible for causing the disease. But that is not the case in most of the solid cancer. Because when you say actually CML, that is called liquid cancer or the blood cancer. So that is cancer in blood cells. But when you look at the cancer in the organs, like prostate, breast, lung, or liver, or any cancer type, the chromosomes are not as simple as the CML. Okay, the number are completely chaotic. Like the one that I showed here, the top, there is a liver cancer, and the bottom is the breast cancer. Just look at that. Here, the numbers are not 46. Particularly in the breast cancer, there are about 100 chromosomes here, and none of the chromosomes are normal. Okay, each chromosome, because this is actually analyzed by what is called the spectral karyotyping, which Dr. Meena Augustus actually she mentioned this morning. So this is just chromosome painting. You can recognize each chromosome with a different color. If the chromosome is recognized with more than one color, that clearly tells that there is a breakage and joining. So like that, you are seeing hundreds and hundreds of these translocations. So now our challenge is to find out which is responsible for causing cancer. Okay, that is the biggest challenge here. It is very difficult to find out that the same kind of chromosome translocation occurs between different patients. I think that is another biggest challenge. Every patient is different. And moreover, even in a single patient, every cell is different. Okay, although because what I'm showing is just only one cell. If I take another cell, I may not see the exact number of chromosomes and exact changes. Okay, there will be some common changes across many cells, but every cell is unique. That is the biggest challenge in most of the solid cancers, why we are not able to actually find a cure or controlling this metastatic cancer, which I will do later. So there are kind of tests.
Just a small reminder about the time.
How many minutes I have?
According to the schedule, you are a little past the time, but if you may like quickly.
Okay, let me go through within like give me 5 minutes. I will just quickly go through that. Okay.
Okay. No issue.
Okay. So recently, there are methods are developed, and I got actually 2 patents for this is called methods for analyzing chromosome translocation in cancer, like that. So the other important point that I want to quickly make is that when you go to the solid cancer, this is the situation in lung cancer. So when you take lung cancer, there are about 13 different mutations are identified. All these mutations are not present in one patient. So one mutation can be present in each patient. So you can classify the patient into distinct molecular subtypes. With all these mutations, there are some drugs available for each patients like that. So here I will simply show you, when you take the initial biopsy, you have to actually screen for all the known mutations. And if the drug is available, just treat the patients with that particular drug so that you will get the benefit. If there are no drugs available, so the patients go for a kind of standard chemotherapy or radiation or something like that. If the patient is not working with any of the drug, they have to do the rebiopsy, then do all additional sequencing, and then to identify what are the kind of additional treatment that can be done. So this is the current status of the cancer screening in terms of kind of personalized medicine or the precision medicine. And these are the different drugs that are developed targeting different mutations. And recently, as you're all familiar, that I think cancer immunotherapy is an emerging field that is based on, there is a Nobel Prize that was awarded to these individuals, which is a major breakthrough. It's all based on the analysis of the DNA. They identified the different genes, like the PD-1 that is more highly expressed in this cancer, which is the roadblock for the immune system to recognize the cancer cells. When you are releasing the block by suppressing the PD-L1, our immune system attacks the cancer cell. So that gives the benefit of immunotherapy, which is a remarkable discovery here. So quickly, I will show you one example. I think this will be more interesting. What is the current status of the cancer treatment? Okay, so even if you have a mutation and a drug is available, you can actually treat the patients. Here, an example, one patient who has a melanoma, who has a cancer all over his body. He had a mutation called BRAF, and there was a drug developed at the time. When the patient was treated with this drug, his cancer was completely cured within 15 weeks. Okay. Then after 3 or 4 weeks, what happened was the cancer came back in the same patient. So what it tells is that, like I told you before, all the cells are not the same. Only a certain population of the cells carry the specific mutation, and the drug will work only on those cells. Then the other cells that do not carry the mutation, they will take over. Okay. So when because there are many subclonal population of cells, and I think that is the greatest challenge, in the cancer treatment. Why some people, they respond initially to the treatment, then they will come back with cancer after 6 months or something. So this is the current status. So again, we have to do more kind of DNA analysis on these patients who develop this resistant cancer type, and to identify any new mutation, the new drugs have to be developed like that. Okay. Then I will quickly go through. Maybe I'll skip these slides. Lessons of time. So I think last 2 or 3 slides. So this is the summary of showing these are the different type of actionable mutations that are identified across cancer. Like particularly the gene fusions, there are about 314 gene fusions that are most commonly occurring across all the cancer types, for which some of them the drugs are available. But anyone who is interested, please go through this link. You'll be able to get more information about this. And then the new development recently, what is going on is called the cell-free DNA. Instead of actually diagnosing, this is for early detection of cancer, right? Like I showed you, the early stage, the cancer do not show any symptoms, so that even the apparently healthy individual, they can get screened just by giving a blood samples. They can isolate what is called the cell-free DNA. I think even it is used for kind of prenatal diagnosis of several kind of clinical syndromes. You can identify some of the cancer-causing mutations in the cell-free DNA. You can tell the patients even before they develop advanced disease, you can start treatment like that. And recently, there are some microfluidic devices have been developed to capture the cancer cells from the peripheral blood. So here, this is the experiment we did in our sample. As you can see, this is a microcapillary You can run the blood through this, and depending on the size because the cancer cells are larger in size and the blood cells are smaller, they can escape through the hole, but the cancer cell, because they are larger, they get trapped in the well. So you can count the exact number of cancer cells, then you can isolate DNA from those cancer cells, then identify the mutation, what is going on. So this is the easy way to capture cancer at the early, before it goes highly metastatic and other things. So this is the new development that is going on. Then these are some of the new developments that is going on in this field. So there is a huge effort in the US that is going on. They are collecting DNA sequence from about 1 million people. They are going to follow up all these individuals, for several years to see how, if they have any kind of genetic variations or single nucleotide polymorphisms, how they are susceptible for any kind of different type of genetic diseases, so that they can develop some methods for early prevention, other things. This is a huge undertaking going on. Then another major breakthrough recently is called artificial intelligence. Using the large volume of data, just you can actually you can send the pictures and they can actually diagnose any kind of congenital malformations or genetic disease. This is called artificial intelligence. Using the large volume of data, just using the facial recognition software, they can tell what type of kind of cancer or the congenital malformation going on. So this is an emerging field that's going on. Even I think here, you don't need the DNA, just you need to have a picture, but don't post your kind of profile picture on the Facebook and other things. People can recognize what is going on your face like that. So I may not have the time to show the video because you already gave me the warning. So I want the students, if you have time, please go through these 3 videos. So when you go to the YouTube, I think you can take a screenshot for the link. So what are the kind of new bold predictions using the human genome within another 10 years? What are the kind of new development that is going to happen? So this is the kind of experts from various fields. They gave this talk. It's about all the 3 videos. It goes for 3 to 4 hours. There is a really very important information is there, more than what I presented. So these are all the bold predictions with the human genome. So with that, I will stop here. And if you guys have maybe time for 1 or 2 important burning questions, I will answer. Otherwise, yeah, thank you.
Yeah, thank you, Professor Nalasivam, for a wonderful talk. Really, it is a highly informative and interesting talk. So it is a good blend and mixture of what happened in the past and current trends, as well as future as prospects, what is happening in cancer therapy, and also what is Human Genome Project, and what is the post aspects, the prospects of Human Genome Project, and various issues like gene tissue expression projects, as well as OMIM and personalized medicine, and really very good and informative, especially for the persons from the other disciplines of the genetics, other than the genetics and all. Highly informative. I will appreciate you, sir. Now the session is open and we can take up question and answer sessions for a briefly for 2 to 3 questions. So now over to Prakriti.
Thank you so much, sir. So the first question is from Pritisha. As we know that the COVID-19 disease is spreading like wildfire, the mutation of the virus is seen frequently. My question is that how genetic testing can give us idea about the mutation within a short period of time so that we can be able to control the pandemic?
Yes, I think that's a more relevant and timely questions. So basically, since we have the complete sequence of the virus, so we know the complete the amino acid structure, what is going on from the wild type versus the mutant versus, okay? So when the mutation occurs within the so-called, the domain called the spike protein, okay? As long as the mutation is affecting the structure of the protein, okay? So that's where the variant is called, because there may be so many mutations that can occur. So what we call as synonymous mutations or non-synonymous mutations. What is called synonymous mutation means that mutation may be there, but it may not affect the amino acid sequence. As you know, when you actually study in your class about the degeneracy of the code, a single code may kind of code for multiple amino acids. So even if there is a change in the DNA sequence, it may not affect the protein structure. So in that way, so people simply, they can identify it as a variant, but it may not actually change much. So that's why whatever the vaccine that is currently developed, if the mutation occurs within the same domain, if it is a non-synonymous, if it is a synonymous mutations, the same vaccine may still work. Unless otherwise, the new mutation that completely change the protein structure, if it changes the amino acids outside the protein or outside the region that is not covered by the current vaccine, whatever they develop, then they have to actually develop entirely new vaccine. Otherwise, if the mutation occurs within a particular protein domain that is critical for the functioning of the virus, then it will be okay. That since we have the complete normal sequence of the virus, it is easy to compare whatever the variation that is coming up. That's why they are actively kind of constantly sequencing the viral genome from several individuals, from several populations, just make sure that whether the virus is undergoing mutation or not. If there's a new variant, they compare with the wild type and make sure this mutation is not changing any amino acids. It is not completely changing the protein structure that might warrant the development of new vaccine. Otherwise, as long as within the domain, the current vaccine is okay. I think that's why you might have seen in the news that, okay, current the COVID or the kind of the Pfizer or the Moderna vaccine are still kind of working on the variant type of virus.
Oh, thank you, sir. The next question is, after knowing genetics of corona as it is spreading by the process of reverse transcription in host cell, so why can't we use reverse transcriptase inhibitors to inhibit further growth of corona, which has already affected the patient?
Yeah, I think it is very difficult to do, although actually in a kind of conceptually it is a novel idea, you can do that. But when you do this kind of blocking reverse transcriptase, how specific you can stop only that particular protein, right? It may affect other normal kind of genes as well. So you may have several non-specific or off-target effects, which makes it complicated. Because that is the main reason even many cancer drugs, when you give a cancer drug, it is not only kind of targeting only the mutant cell, it affects some normal cells as well. That is the main reason when patients are undergoing chemotherapy, you have like a lot of side effects like losing hair and other things, So these are all the off-target effects. So unless you have a specific, kind of targeted effect, you cannot actually use this approach to stop the reverse transcription only on the virus, because it may have some off-target effects and give some non-specific kind of symptoms and other things. Even with after all the clinical trial and everything, some individuals, it is not effective as well. So I think because of the unwanted off-target effects, it is very difficult to control in that approach. But conceptually, it is a novel idea. Yeah.
Thank you, sir. As we are running out of time, we would like to mail some questions to your email, sir.
Please, yeah.
And as we come to the end of this session, I would like to thank our keynote speaker, Dr. Nalasivam, for enlightening us with his knowledge. Today's webinar was full of knowledge and interesting things. It gave us deep insights into the topic and also revealed some interesting facts. I'm sure that this lecture by Dr. Nalasivam will help us in our studies in future. Once again, I would like to thank our keynote speaker for taking out time from their busy schedule and being a part of this event. Thank you so much, sir. I would like to thank our Dean, Dr. V. Pragasam, for encouraging us to conduct this webinar, our HOD, Dr. Asha Devi ma'am, and our event coordinator, Dr. Radha Saraswati ma'am, for being a constant support in organizing this lecture for all of us. I would like to thank the student committee who worked really hard to make this event successful. Also, thank you to all the participants for being attentive and learning. A special thanks to our session in charge, Professor Bhaskar Rao sir, under whose guidance we could make this event successful. Thank you.
Thank you, Professor.
You are talking longer than me.
Nalasivam.
It's okay.
Thanks a lot. And it is very, very informative. A lot of things I also learned from this. Thank you very much for your time.
No problem. Please feel free to kind of send your questions to my email. I'll be happy to talk to anyone.
Yes, Professor. Yes, Dr. Nalasivam.
I will get in touch with you with the rest of the questions that are there.
And also, you guys have my phone number. You can actually do some WhatsApp chat via WhatsApp. I'll be happy to talk to you guys.
Sure, sir.
Okay.
Thank you. Good night.
Okay. Have a nice rest of the day. It is 1 o'clock for me. Let me go to sleep. Yeah.
Thanks a lot for your time. Thank you.
All right. Thank you.
Professor. Thank you, Dr. Nalasivam.
Ma'am, shall we begin with the next workshop? Gomti sir?
Yes.
So, shall we begin?
Can you hear me?
So the voice is echoing a bit, sir.
Now?
Still there's some noise, sir.
Yeah,
Maybe it is running in two devices. You
Yeah,
can
yeah,
off
yeah.
one device.
Now?
Yeah, so
It is
better.
open. in-
Yeah.
Alka ma'am?
Yes, I am here.
Yeah,
Can
okay,
you
ma'am.
hear me?
Yes, ma'am, yeah.
Yeah.
Shall we start
Okay.
now?
Yes, please go ahead.
Sure.
Ma'am, can switch on
morning.
your video.
Yeah.
I don't know, it is not switching on. I'm trying.
We'll help you. And Gomati Nayagan, can you please switch on your video, please?
Yes, ma'am. I think I already have on one of the devices.
Yeah. Okay. Thank you.
Yeah,
Shall I
mine
begin
is
now?
not. Yeah, Vaishnavi, start please.
Yeah. Okay. Good morning, everyone. I, Sakshikara, along with my co-host Tama Meda, welcome everyone for today's NGS analysis workshop. We are very glad to have so many enthusiastic participants among us amidst the pandemic situation. A special thanks to Dr. Alka Mehta ma'am, who has taken up as the session in charge for today's workshop. A very warm welcome, ma'am. Thank you for your presence and guidance throughout. We are grateful to have you among us for today's workshop. We would like to begin by giving a brief intro about Gomti Nayagam sir. Sir is a budding entrepreneur in making biotechnology products smarter and affordable. Completion of his Bachelor of Technology from July 2017 to May 2015. So completed his MTech by research from July 2017 to July 2020. So briefly worked as an HPLC GC analyst from June 2016 to September 2017 and was also looking after the stability study of drug substances using Novalens. Apart from research work, Sir offers lab classes for UT students and is the operator for flow cytometer instrument in our very own core facility, that is Vellore Institute of Technology, and also as a teaching cum research assistant from November 2017 to November 2019. Looking into the publications by Sir, which will be briefed by Medha.
Thank you so much, Sakshi. Talking about the draft genome sequence of highly pigmented bacterium, which is Paracoccus mercusi, KGP, capable of producing galacto-oligosaccharide synthesizing enzyme. Amelioration of doxorubicin induced cardiotoxicity by oral lyophilized Duna-Liela supplement in a female Vista rat model with active estrous cycle and surgical menopause. Metagenomic analysis of the fecal microbiome of rats with 1,2-dimethylhydrazine induced colon cancer and prophylactic whole cell carotenoid intervention. The special award was for Summer Research Fellowship 2013 for project learning and overexpression of recombinant AABT AMP in E. coli by INSA, IAS, and NAS. He did his internship of 6 months in the year 2015 from MSD Wellcome Trust Hillman Laboratories, New Delhi. Another internship of 6 months from November 2015 to May 2016 from Biocon Pharmaceuticals. Sparking in areas of interest like NGS analysis, metagenomics, bioprocess engineering, gut microbial ecology, and cancer biology. Holding up key skills of whole genome sequencing and analysis, transcriptome sequencing and analysis, small animal handling, and flow cytometry.
We are very grateful to have you with us today, sir. A small keynote to all the participants. Please mute your mics. Any queries can be posted in the chat box. We will make sure that your queries are answered. We hope you all have an amazing experience and gain all the knowledge you can from it. Before beginning with the workshop, I would like to ask Alka Mehta ma'am to address the audience. Over to you, ma'am.
Good morning, everyone. Can you hear me?
Yes, ma'am, yes.
Yes, okay. I welcome you all in the first workshop on the next generation sequencing, the NGS data analysis. As we know, the DNA sequence is the blueprint of our life, And Sanger sequencing has revolutionized our knowledge about the genetic makeup of the living being. The NGS is the next step in this direction and the technique of present. It facilitates whole genome sequencing quickly. It utilizes the RNA sequencing. It can analyze the epigenetic factors, facilitates the studying the microbiome. Even it helps in early diagnosis of the cancer and so on. So sequencing is not a big deal as it is carried out by a machine. Real challenge is to analyze the sequence and get the desired information. And so I invite Mr. Gomti Naigam to teach us how to analyze the NGS data through this workshop. Welcome, Mr. Gomti Naigam.
Thank you, ma'am. Thank you very much. And I thank both Sakshi and Mameta. I also take this opportunity to thank Dr. Radha ma'am for giving me this opportunity. So without much ado, I think we'll begin with our workshop. So I'm sorry about the video. I'm a bit away from the light, I think. So I couldn't do anything immediately about it. So shall I start sharing my screen?
Yes, sir, please.
So yes, welcome to NGS Data Analysis Workshop. This is workshop 1. I'm incubator, actually pre-incubator. I'm in the process of registering my company, and also it is pre-incubator at the ITTBI. So this session I have planned for 1.5 to 2 hours. So are we going to touch upon general basic things about genomic analysis, microbiome sequencing and data analysis, along with transcriptomics. We'll touch upon how it is done, how it is begun, and then how it is processed out. So also I'll introduce you to genomic data file types, what are the different file types and what to do with them, how to do analysis with them, if you did not know already. And also I will tell you about basic Linux commands to operate for this particular workshop. So I hope all of you have got this Ubuntu distribution on the Windows system, or if you have already Linux installed in your PCs. So could you say a quick S at using the chat box? You hear me? Yes. So yeah. So I specifically like Ubuntu. You see, I'm sure you might be knowing the meaning of the word Ubuntu, I think. Ubuntu means I am because you are. Ubuntu means oneness. So that's why they named this for open source Linux distribution. This is an open source Linux distribution. Anybody can go and contribute to it and make changes to it and make it as effective for themselves. Means like it is customizing it. So the same thing I expect you all to do here. So if you have run into problems, you can please let us know through the chat box, and the other friends quarrel in the forum could help them out. So this is what is I am because you are. Okay, could you please help this out? So I'll also help in setting up the basic environment for genomics analysis, and I'll provide you with some tools and some basic data, some minimalistic data to begin with. So this has been designed for beginners in both Linux and in this data analysis. And I'll walk you through the GitHub repositories and have planned few other tools to graphic user interfaces and other open source tools for you. So any sample it starts with collection, sample collection. You have to make sure that it passes a particular quality control. Then you prepare a sequencing library. Then you make choice of your platform sequencer, like Dr. Alkameta told this. We can put through the sequencer, and the real challenge is next to it. We have to analyze the data. We need to understand what is there in the sequence of DNA. Also, we had this morning we had Dr. Nallasivam Pannaswamy speaking about Human Genome Project, and beyond Human Genome Project, personalized medicine, what are all required for that. And this is like a linker between those two things. We have got the DNA, and we are going to look into it we're going to understand the problem from it. If it is for clinical aspect, yeah, if there is any mutation or dysregulated gene expression, anything like that, we'll understand from it and we'll make a decision, clinical decision based on that. So I'll skip most of these slides, I think, because I'm already out of time. So first step in sequencing project is, what type of sample you're going to work with. Maybe you are interested with some bacteria, some mammals, or you might be interested with environmental DNA, like you're looking at the fecal content, and if you're looking at sediments from Antarctica, Arctic, meteorite, or even if in the future, near future, we'll be looking at Martian soil also. We'll look for life forms if there is any history of life forms in that. And also there are present advancements, current recent advancements in transcriptomics to study the gene expressions through RNA sequencing. So, and after that, there is this very recent advancement like sequencing the RNA from a very single cell. Okay, so I'll talk about it. And for that particular single cell RNA sequencing, we require a sophisticated instrumentation, which is called as FACS, fluorescence assisted cell sorter. It is an appendix instrument attached with flow cytometry. So we need that. We have to isolate the cell of interest. From that, we have to isolate the RNA, and we do complementary DNA synthesis, cDNA synthesis from it, and we sequence it. And also we could do, like, we can directly sequence the RNA. There are platforms available to do that. So coming to the eDNA, it is called as microbiome studies or metagenomic study. Metagenomics means meta, multiple or complex DNA coming from the environment. It might include viruses, bacteria, it even includes some eukaryotic organisms to understand what chemistry is present there, and some other information, and I'll show you the information later. So I'll touch upon the sequencing technologies like sequencers, because we need to know about the sequences a little bit to know about the sequencing data, and only then we can proceed into the analysis part. So after a while, I think I will turn off this video if you don't mind, because of the bandwidth restrictions. I'll be downloading some data over it, and I'll be showing the terminal window. So after this presentation, I'll turn it off. So as you all know, I probably might have heard about standard sequencing, because I see most of you are from MSc background or BSc, something like that. That's the first generation sequencing tech, Max and Gilbert method also. And the recent one is Nanopo sequencing method, Pacific Biosystems, and one more is coming up, which is called as DNA Nanopo sequencing. And there's a gold standard sequencing method called Illumina, which is widely called as NGS. Also, there's another name called sequencing by synthesis. So there are other methods as well, biosequencing, ABS, these are different platforms. So different platforms have different pros and cons. For example, Sanger is not cost effective. Illumina is cost effective and accurate, so that's why it has taken over. And there are recent advancements; those are still more cost effective, like nanopo sequencing, but they are less accurate. But still, there are some other advantages over Illumina sequencing. So we'll get into it. These are introductions. We can even share this PPT with you if you want. So I'll mostly skip through this, I think. So you know what happens with Sanger sequencing, I hope. So it is the dideoxy nucleotide base pairs, which is called as chain termination method. It goes and binds, and where it binds, it stops. The extension of the DNA stops there, and it is run through randomly. It stops there, and it is run through capillary electrophoresis, and this is how it is decoded. Where it is stopped from there, using the fluorophores, it is detected. The capillary electrophoresis could be assisted with a laser source. It excites the fluorophore in the dideoxy nucleotide base pairs. G would be having a different color. C would be having a different color. So based on the color, it is differentiated, and it is sequenced. Means it is inferred back. Okay, using computational methods. So the similar side means similar data was presented by Dr. Nalasin this morning. Yeah, he told that cost of per genome sequencing would come down to less than $1,000, and it has already started to come down very well. You could even do even less than that using Illumina platforms. So this is the data until 2014. It is still improving day by day. So after next generation means standard sequencing, I'll touch upon Illumina sequencing methods. So this is what happens actually at Illumina method. So there's two different, there are many important things to look upon in the Illumina method. I'll talk about two things, pattern sequencing and a single end sequencing. So this is the pros and cons. This is called as, otherwise called as short tree sequencing. And this Sandwich method is called as long read sequencing, as well nanopore passive batch systems are called as long read sequencing methods. This is a short read sequencing method. This has given us a disadvantage, but this comes with an advantage actually, because it is called a sequencing biosynthesis. It uses high fidelity PCR polymerase enzyme. So as short as it is, so it can be more accurate. So that's the reason why it is like that. So I talked about this. This is what already I showed you. We get the genomic DNA if we are interested with sequencing genome of an organism. So we get it, we fragment it. Already this Illumina could be able to do only 150 base pairs. So we need to fragment it. We have to choose only 150 base pair fragments from it. And we attach adapter sequences. Adapters are also some sequences, specific sequences. So it is attached to them so that the adapters are complementary sequences to the sequences which are embedded on a semiconductor plate, which is called as flow cell. It will be there. I'll show you later. I have a different platform, nanosequencing flow cell for you. I'll show you later. So this adapter goes and binds to the plate, which is a flow cell, and it holds the DNA for it. And we do this bridge amplification. This particular image shows about the pattern sequencing method. So at the other end, there's a different adapter which is complementary to the adjacent one in the flow cell. So it forms a bridge, it goes and binds to it, and now the PCR sequencing is done. Sequencing by synthesis, it is what it means. It is done, and then a very large number of clusters are formed from it. It will be done for many number of cycles, and a large clusters of the same sequences will be formed. And the same way, the fluorescently labeled ATGC will be made to flow through the flow cell. That's why it's called as flow cell. It will be made to flow through, and if we have found a complementary nucleotide base pair in the sequence fragment, so this respective one would go and bind and would give that specific light. For example, here is given as A bears red light, C blue, G yellow, and T green. So from this flow cell, from the top of it, the SCCD would record it completely, means for every cycle of flow of nucleotide base pairs. So it will record every color, and these colors will be stored like how it is done during capillary process, and later fed out into the computer, and the computer will do the processing. This is called as base calling. A raw data in the form of colors will be given to the computer. Then computer does some basic processing, which is called as base calling, nucleotide base calling. It calls the nucleotide bases from the color which it has obtained from the sequence. Okay, this is what it is. So this is an image of the Illumina flow cell, and then we also have got a flow cell of nanopore sequences. It would typically look like one. So this is the flow cell of nanopore. I think you're able to see it. I'll make it a little more closer. The one which you see here, this is the flow cell particularly. This is lipid handling systems, which are defined. The sequencing is happening only in this small chip. Next slide, we'll see how nanopore sequencing works. Okay, so please remember this. So nanopore sequencer is the smallest sequencer ever found, which is just a size of a stapler. If you wish, I could show you that also. One here. So this is the size of an entire sequencer. You see that? So this is a symbol of nanopore, Oxford nanopore. So this is where we keep our flow cell. This is where the flow cell is attached, and we connect this sequencer to the computer. This will read this in a similar way. We'll see that in the slides. So here you see there is this flow cell map, which is a semiconducting membrane. To that, E. coli porine proteins are embedded on it. Okay, so a constant current will be flow through the semiconducting membrane here, and there will be some capture proteins called as rotor proteins. So they capture the DNA fragments and they let it pass through this porins. So as the porins through the porins, as the DNA passes through the porins and it cuts the current flowing through the semiconducting material, some kind of.
Now it is okay. Okay, I think.
Sorry.
Audio is okay. Just I will check video.
Okay. Yeah, as the DNA flows through it.
Yeah, mic is open, but this video is not opening. I don't.
So as the DNA fragment flows through it, there will be resistance created. That resistance will be recorded by the computer, and this will be again called by the somewhat a different computation process. So this is about it. So we saw about different technologies, how standard sequence and the Illumina sequence the DNA fragments, and we also saw how a nanopore sequence it. So this is an example of a standard sequence. This you could find in NCBI also in the repositories. So here you see this is a FASTA file. Actually, this is a FASTQ file presented as a FASTA file. FASTQ, FASTA, the difference between them is the Q. The Q stands for quality. The quality of this every base pair. Okay, so we will see about it in the later slides, coming slides. So as you see, the pink color one or low quality, you could see that here. The yellow one are in the median range of quality, and if it is green, it's of very good quality. Very good quality means very good confidence. Very good confidence that it is T, the confidence to call it as T, call T as T and G as G. Okay, so this is how a FASTQ file from Illumina would look. So it starts from author rate. It will have a header following the author rate, header of from where this GD was given, how this was barcoded, and such information would be present there in the header. It's a information like a name. And the next line would be the nucleotide sequence, the sequence of the DNA, and it ends with a plus symbol, as you can see there. And the next line starts with some symbols. These every symbol corresponds to a particular quality score, which we will see briefly. So this is what I was talking about. So each symbol stands for a particular quality score, and this is how a quality score is determined. So if a quality score of 10 means only 90% of the total base pair sequences is of confidence, good confidence, means only 90% could be accurate. If you sequence 100 base pairs, only 90 base pair is confidently them. Other would have happened by error. Okay, so say likewise, you can see that here 20 and 30, 40, likewise here. So the maximum is 99.99%. This particular thread score is used for nanopore sequencing, and there are two different thread scores, thread 33 and thread 64 for Illumina based and nanopore sequencing. So this formula, how this is calculated. So I think we can begin setting up Linux environment right now. So this is quite important information we need to know about. 99% of NGS data analysis happens only in the terminal environment. It is a command line interface. You see only a terminal, not a graphic user interface, like we say in Macintosh and Windows. Ubuntu has a graphic user interface, but most of these tools work in command line interface. You will see the difference. I'll tell you how to find the difference, means why it was in command line rather than in graphic user interface later. Okay, so there are tools for graphic user interface also. I will introduce to some of those tools. To the left side you see a similar one. The right side you see CLI. So which one do you feel is comfortable? If it is CLI, you type C. If it is GUI, you could type G in the chat box. Could you let me know quickly? So, okay, yeah. Naturally, many would find graphic user interface as more comfortable than a command line interface. Okay, yeah. So we'll see into command line interface today, and also we'll get into a graphic user interface. So these are the common commands we're going to look into. Now I think I'll turn my video off. So people who are more familiar with Linux environment might be knowing it already. And if you do not know, so learn these basic commands. So I think I'll start the. So all of you could start your WSL environment, I think now. So are you following it? Have you started the could you say could you confirm by telling a quick S if you have started WSL? Okay, so people have started. Good, thank you. What do? You start WSL. I think the program coordinators and panel members have sent you some instruction how to set up a WSL. In case few of you might be having a problem in setting up this, if that's the case, I give you one more way to do it. That's called as onworks site. Onworks Ubuntu. You could set for onworks Ubuntu. I'll type that out in chat box. Go to this website, onworks, and I like Ubuntu because it is more friendly and easy to use than any other Linux distribution. Likewise, Mint also would be easy. Set for the same in Google, and you would see something like this. I'm sharing my screen right now. I think you're able to see my screen. So look for Ubuntu 20 if it is comfortable, or else you could even use version 16. It's there. So, but you can click on it and click start, and you have to wait for some 5 seconds later to this, and you have to again click start, and it will start an environment for you. So if this is for people who are facing problem with only presentation is visible, okay, thank you for letting me know. So now do you see? Did you confirm? Yeah. We would work on terminal. For those who have not set up this WSL, will go to this on website and they will switch to terminal here. You find terminal? Yeah. The password, the default password here is 123456. It might ask for some password later. Okay. Once you've got this, I'm turning it off. I'll move to my WSL in my laptop. Okay. Shall I? So I hope all of you are following me. Could you confirm by telling a quick S, alphabet yes? Yeah, thank you very much. Now I'll share WSL. So do you see my screen now? My terminal window? Could you confirm? Okay, so shall we begin? What I'm going to do right now is I'm going to install a tool, which is an assembler tool. And after that, we'll go back to the presentation. We'll see how that works. Okay, we'll see how a faster file would look from a faster file from standard would look a faster file from Immunotherapy. Okay, so now you follow me carefully. I would put the same into the chat box as well. Sudo apt get also you could use. I'm using apt install. Sudo apt install. The software which we are going to use right now is Spades. Sudo apt install Spades. Okay. You would have set up a password for your computer. I'm just typing that now. You wouldn't see the password now. Okay. No problem with this package. So this is the command. You have to type it over there. So this should work for most of you. I'll come from a different computer right now where it should work. Okay. So now I think you're able to see the screen. Yeah, if it has started installing, very good. Okay, so I think I do have it already, so there's no other reason to install it again. So started installing. Once it is done, you let me know. We'll switch to the presentation. Okay. So everyone able to follow me? Not installing. What's that error you see there? That is, could you tell me the exact error there? Yes, you should type the same command as mine. I'll repeat the command here. Pseudo install space. It says unable to locate package for somebody. Maybe you should update it. Sudo type sudo apt get updated. So have to update your environment and you try later because mine the other terminal was a newly installed one. It cannot work. Yes, you should type the command in the browser. So if you still face some problems here, you just simply follow me doing it. So I'll tell you how to report those errors to me later in time. It would be open for a week or so. So you could follow me simply right now, and you could try that later. Okay. Just but let me keep letting you know, keep posting about the link I would share in the meantime. Okay. Now you see that if you see, I think most of you have got space installed. So could you confirm by telling yes? Still unpacking? Okay. For those who are not able to get this Spades package, yeah, I'm posting it in the chat right now. You follow the same commands there. Okay. I have given wget followed by URL. Do you see? If it says unable to locate package for people, those who get this error, you just follow me. I've just posted a command wget followed by the link. You could copy paste it into your terminal right now. So this should install Spades. It should collect Spades. to your means speech binary files to your computer, to the computer. Is that happening for everyone? Yes, it is getting installed in your WSL environment. Yes. So after that, once if you have got it, we have to unpack it. I'll just copy paste the means I'll put a command on it. You just copy paste it to your terminal. I'll put the same on my computer set. So just ensure that it is the same, yeah, same version. So it's unpacked right now. I'll put the same in the chat box. Yeah, you do this. Tar followed by -XZF space. No, so still going. Okay, it is still collecting the package. Okay, it depends on the network and the speed of the computer. Okay, let it happen. No problem. We have time. We are going to shift to the computer right now. Once you are done with this specific command, tar-z means unpacking it, I would want you to do a quick check by typing ls in it. Okay. If I, for example, if I type ls, it will show the, it will list what are all present in my computer right now. Okay. So as you see in the screen, I have a compressed package, a gun zip package of space, and also an unpacked one. Okay. So the unpacked folder would appear in blue color for you. So cd space. I'm typing the same to look into what is inside that. You should find a binary folder. So I'll move into the binary folder. I'll list it back again. So we'll see some tools like this. Okay. You will see all a Python. Okay, you want the command back again. After WK, you have to type. You have to unpack the compressed package using tar-z -xzf followed by the name of the package. Okay, so I think you're all able to follow me until now. So for most of you, it should have worked. The previous sudo apt-get install page should have worked. For those who did not work, follow the command. So let it run it from behind. So I think we'll switch to the presentation now. I think it's still going on for a few. So we'll go to the presentation right now. So here I would introduce some of the terminologies involved in the sequencing analysis. You know what is DNA sequencing, what is a genome it is, what is an RNA sequencing, what is a transcriptome, what is a read. Read means there are raw reads coming out of the sequencer. Okay, that is mostly a raw. We will make use of it. You know what is amplification already. Okay, these are very basic things. So now, is everyone following me? Could you confirm by telling a quick yes? Yes, sure. This can be run in Conda environment. There's a Conda package, Conda speeds package. You could do that in Conda also. You could find that easily. Thanks, okay. So if anybody having Conda environment installed in their Linux computers, you could use Conda packages of speeds. Okay. So is everyone following me right now? May I move to the next slide? Could you confirm by telling quick yes? Debian, fine, no problem. It's still a Linux distribution, no problem. Oh, it says complete, but no app is installed. You couldn't find Spades. I think Spades should work for a Debian environment also. You type where is Spades in your terminal and just let me know what it is showing. You just copy paste it output here. You should not use Control C or B on the terminal. You should use right click to copy from the terminal and you paste here. Okay. So shall I move on to the slide now? Following me, anybody? Shall we? So it means it is already there. Ms. Shivani Iranki, I think it's already there. So you don't have to worry. You can use it directly. Okay. Now what you do, you type space and just copy paste the output of the terminal here. So I think now it's time for me to move on to the presentation. So these are the things which are done to process the reads before it is put through an assembler like space. Okay. Still there are queries. So where are you confused about? Could anybody help Prakati out? Prakati, just tell the error which you get into the chat box. So maybe people will be able to help you out. Okay, so we saw while preparing a library, we attached adapters to the DNA fragments. And also sometimes we would also attach some different kind of sequence called as barcodes to it. Barcodes are nucleotide sequences, but they are like labels. How a barcode is attached to a product in a shopping complex. These barcodes are attached to the fragments just to identify from which tube this came. Suppose if you have two different sample tubes, two different genome URL, let's say, you will add barcode 1 to one kind of tube and barcode 2 to two kind of tube. So this barcode 1 and barcode 2 will be of two different nucleotide sequences, which the computer can understand, okay, which these tools can understand, and it will understand that after you mix up these barcodes, you could pool these two samples together and run it in a flow cell. Just as I showed you the flow cell, everything will run in the same pool, okay? So at that time, we will not be able to know from which tube or which sample this came. That is why these barcodes are in place. So they label the DNA fragments just to infer from which tube this came. So while we process it, we'll come to know it's from this genome of interest, and these sequences are from a different genome of interest. Okay. So while assembling it, we need to remove these adapters and barcodes from it. The way you pool together these samples are called as multiplexing. So if you want to segregate these cells back, you have to use a demultiplexing tool, which would come mostly native tools are present there to demultiplex for these things. And just as I told you, there are apparent reads and a single reads and long reads. These are all we are going to look at right now. Okay. For apparent reads, like I said, one sequence would be read in means the same sequence would be read in two different ways. One is forward 3-5- and again another read in 5-3-. So this during mid-pair merging, these two will be overlapped, so that if there are any discrepancies between these two forward and reverse reads, that will be removed. There should not be any discrepancies. That's why to identify it, there's a pattern sequencing, so that we will get a more accurate read for it. Okay. So there's a mid-pair merging tool called as PEAR, PEAR. I'll put that in the chat box, so you could follow it later also. Here, you could look for this PEAR tool. Okay. So we don't require this tool right now. So it is required, means it requires some kind of registration to use it. Okay. So to remove the adapters, there are tools like CutAdapt and Trimming. Trimming is trimming the sequence ends. Okay. Trimming is used. De-indexer is a kind of a demultiplexer. Okay. It will identify the barcode sequences and put it on a separate folder for us to identify from which tube this particular genome came. Okay. So now we have got all the reads purified from adapters and trimmed, and now we have it de-indexed, and now we need to check the quality of it, the quality of the sequences using a tool called FastQC. There are some other tools, but this FastQC tool is more popular, and I have just put output of that FastQC file in the next slide. As you can see, it will give you a summary report of it. It will predict, give you basic statistics. What are the length, average length of the sequence reads in it, and the per base sequence quality, like a Fred score, the GC content in it, the Kramer content in it. Is there any duplication, and is there any anomaly present in it? Okay, so this is what a FastQC file will do. I think there's some problem for Miss Preeti Shah. I think. Okay, is that means you're not able to follow it? Have you installed Spades? Could you say yes? If you have any problems, if you're stuck with some step, you could please post it on the chat box. The friends must be able to help. Means the friends in the forum must be able to help it. You couldn't follow it. Where are you stuck? Two slide. Okay. Means it's not a problem. Anyhow, if you make a request, I can share it with you. Just share the entire slide with you. So no problem right now, because already I'm running out of time. I think I'll make it fast. Okay, Pritisha. So we see all genome assemblers working. Okay. Now I want you to know how an assembly works, what an assembler does. Okay. Are you able to see the screen now? Everybody, could you please follow me to the slide? Could you confirm by telling a quick S? Are you all following me? Yes, you're all able to follow me. Okay. So you're on it. Okay. So suppose, just assume that these are the reads we have got. Okay. I'm going to give you a task now to decode what these reads mean. These are the reads which is going to mean a word, a lengthy word. Okay. Could you try finding it out? This is what basically a sequencer does. There are two different types of assemblies. One is called as de novo assembly. Other one is called as reference-based assembly. I'll introduce you to that as well later to this. Okay. So I think it's visible to your screens right now. You see CES, ation, trans, terrific, TRA, Cester, and CES ion like that. So could you decode what does that mean? My voice not clear. Is it clear right now? Renathan, could you confirm? Is it clear right now? Yeah. Thanks, Saket. It is transesterification. Yes. It's echoing a bit. Okay, I'm not sure. It's because of the room, I think. Okay, I'll try the best. Okay, yes, it is transesterification. Saket has found it. So this is how a reference assembler does. For a reference-based assembly, we need to give a reference sequences, which is the reference. Transesterification is a reference given to the assembler. Spades is a de novo assembler. It's not a reference-based assembler. It would match like this, like it does an overlap. It will overlap the reads over the reference, and it will find what does that mean. So I think this particular slide would make it clear for you how an assembler works. So it's a basic way of understanding it. This is how an assembler works. Okay. So are you all following me? Could you confirm by telling a quick S, alphabet S? Oh yes, wonderful. Thank you. So soon I'm going to give you some raw data to work with. Okay. Before that, we'll look into some of the terminologies involved in this thing. Shotgun sequencing. What does that mean? Shotgun sequencing actually means breaking a DNA fragment to smaller fragments. Illumina method is more widely called as shotgun sequencing. And de novo assembly, that's what I just told you. There's a reference-based assembly as well. And there's something called this coverage, okay? And base calling, I told you already. The raw data is converted into a human readable form, into ATGC. That is called base calling. And assembly means assembling the raw reads into it. Annotation means finding what is the meaning of the sequence we have got. So, just right now we assemble a fragment of alphabets as transistor application. Now, annotation means to look for the meaning using a dictionary. Okay, that's what annotation means. Functional annotation means, actually, I should not use a plain annotation. Functional annotation means understanding from a reference, basically. Okay, and you know what is a classifier, and you know what is a classifier, and there is this context, scaffold, and entity value. I will tell you later after assembly. Okay, so I showed you how an overlap graph means overlap-based assembly works there. This is how exactly it is done. Okay, and there is one other method for this overlapping. It's very much similar to overlapping, but this is called a graphing method through Keynote fragmentation. Okay, and there's another method called string. This wavering graph is more prevalent. It is widely adapted by many assembly tools. Okay, suppose that's you at the top of the slide you see a genome sequence, and for that we have to append some raw rates. The same sequence fragmented into different forms. Okay, it is trimmed here and there. That's how a typical raw rate would look. Now, this short fragments of reads, raw reads, are further fragmented into particular mers. Here we, for the example, we see 5 k-mers. Every short read is cut into 5 k-mers, beginning with ACGAT. So 5 is done. Stop it there. Now you go to the raw read again. Skip the first sequence. Go to the next. Start from that CGATG. Stop it there. Terminate there. Now, okay. Now you go again to the first of this one. So you forget the C and you go GABDC. Likewise, this entire Kramer graphing is done. And from this Kramer graphing, this overlap is continued through. So I've given the growing graph example here. So ACG is compared with the CGAT, the next Kramer, and it is overlapped subsequently like that, and the final assembly is produced. Okay, so this is how it would look. The growing graph would look in different Kramer sizes. For Kramer 50, it would be shorter fragments. For Kramer 1000, it would be different. And for 5000, it would be one lesser than that, because size of equally would be around 5 MB. Okay. So I think I'll now take you to the reads right now. I'll give you with raw reads. Let it download. Also, we'll take a quick break. Okay. So you're all right. You're all following me. Could you confirm by telling yes? I'll now share the link to download the data. Okay. This is where you should go. I'm just putting it there in the chat box right now. This is a GitHub repository. I have a repository there, and I have posted some data under the folder genomics, under the repository genomics. So once you get into it, just let me know. You just use this link, open this link in your browser, and you come back to your terminal. Once you come back to your terminal, type alphabet T. Okay. Once you got this, copied this web link, yeah, you should pull the data from there. Yes. If you once you are done, once you move to terminal, you type T. So once you have done it, you type T so that I move. Once we start downloading it, we will break for 5 minutes and we'll be back. Okay. So people started to get into terminal. So we copy the link, earlier link. So if you go into the browser, this is how the repository would look. So are you all able to see this repository there in your browser also? You can, yeah, you're able to see it. Fine then. So also you could post your issues here at this link. To the top, you find issues. Okay. So you could create an account here and post your issues. If I was not able to solve your error here, issues here, you could post it there. It would be open for a week from now, the Saturday to the coming Saturday. So I can help you out with that. Okay. Hope all of you have got that. You can go to this issues, post your issues there. Okay. Okay, great. So hope you all have got this link, copy to it. If you did not do from the chat box, you could do from, you can click this green code box. You could copy this into your clipboard using the clipboard symbol. Just plainly click it. Now, I'll also move to the terminal. Okay. Now, I would ask you to do the same thing there. Okay. CD space tilde. This tilde would be there in most keyboards and just beneath the escape key. Okay. Just type CD space tilde. So it will take you to the home folder. Okay. Have you all done that? If you have done, could you confirm by typing tilde in the chat box? Oh, great. So Yami has done. Megna, yes. So quite good number of people are able to follow it. So I think I'll move on to the next. I'm typing git, git flow, and I'll plainly right click it. You should not use Control C or B in the terminal. It's a common rule. If you're new to the next terminal, and press enter. So let it download. We'll have a quick break now. Okay. So mine is done. It got downloaded for me. So I hope the same will happen to you. If there's a problem, you could put it on the chat box so people would help you out with it. So the time now is 11:25. We'll be back here short at 11:30. So for some, this download might take time. That's why I'm taking a break here. 11:25 to 11:30 sharp. Okay. Thank you very much.
a small announcement. We'll have a break of 5 minutes and so we'll continue back again. Thank you. Yes, sir.
It's not 1S, it's LS. Ria, it is LS. I'll post the same comment here. Okay, so right now you see genomics file here. Is everyone following? Is everyone back to the terminal? You confirm, please. Okay, yes, thank you. So here, right now, I just saw what all present inside my home directory. So I'm just going to move into the directory which I cloned from the repository. Okay, which is CD followed by space genomics, and click enter. So I'll post the same there. You have only genomics written because you have only one tab, only that shared. So when you type this CD space genomics, so okay, once this is done, we will see what are the files present in it. I'll type ls again to see what is there in it. So we're running out of time. Be better, be quick. Okay. You see readme.file and also a folder which must be in blue color for you also. Okay. So I'll move to that quickly. Type SAM and I'll press tab key, so it will automatically capture it, and then I'll press enter. Now I'm posting the same there in the chat box. CD space SAM. You could do with easy capture. I'm typing that sample data. So we'll see what all present inside the sample data. I'm just using ls list command to see what all present there. Already okay. So there is a sample long read, and there are two short reads, I think. Short reads 2 is there. Short reads 1 is not there. Okay, it's uncompressed. Okay. So I'm also better. So what you all do here, you type PWD and press enter. Okay, you should see the pathway where the raw read are present. Okay, you just highlight it using your cursor and use Control C. You could use that with WSF, but with Linux terminal you cannot use Control C. Okay. Okay. Once this is done, we'll move on to assembly section now. So now we'll have a short look at it. Short look at the reads, how the reads would look. I have here these short reads are from Illumina. Okay. So I'm using less command to visualize it. Inside the sample data folder, type LASS and followed by type short. You can use tab, it will stop where it is redundant. Okay, it stops there. And I'm telling to open short reads dot fast one.fastq. So this is what you see. And you see that there's a header which says short read 3 of 1. And you see the sequence, very short sequence. Okay, it ends with positive symbol, positive sign, and there is this validity score. Okay, so this is how a short read would look, and we'll also see this long read, how it would look. Okay, before that we need to uncompress it. You could not follow the means, you cannot do the same thing there in your computer, I think, because it's not decompressed. So I'll decompress the package using Gunset, followed by low. I'm pressing the tab. I'm going to open only the low depth one. Low one, scroll tab again. Okay. Now if I type less, I will means list command. I should see the uncompressed, means decompressed folder. I'm using less to visualize that. Type low tab. Now again I give low tab. That first view. Then I'll press enter. So are you able to see? Are you all following me? This is how a long read would look. Starts with authorate. It has a header. It says long read 1 because that's the first read. And you saw the difference, means you could see the difference easily here, that long reads are this length here versus short reads are only this much. Okay, and it ends there with a positive symbol. And this is the Fred scope. You still can go further into it. Each symbol corresponds to a score to a corresponding nucleotide base pair. Okay, it ends there, and it's the next it starts with authorate with the header long read 2, and the sequence likewise. Okay, now I'm quitting this using Control Z. It says no such payload directory. Maybe you are missed somewhere. Okay, so you type pwd, and just post the output here. Okay, if you type this command, what do you see there? You just ensure that you are inside Genomics sample data folder. Could you confirm by telling S? Alphabet S is enough. Yes, you are inside the sample data folder already. Okay. Okay, fine. You're inside sample data then. Okay, fine. So do you see this these files? Any long reads, high def, reference.fastq, there's a sample context file, short reads that anything like that? Are you able to see it? Could you confirm yes? Yes, you're able to see it. So to exit the long read data, you press Control along with Z. Control along with Z. Okay. So a few of you were able to open it. So now you try less followed by, you have to type the entire name there. So you just follow what I'm typing here. Less you should be able to see that from now. If not, you might be having a G set next to the format. Okay. Okay. If you have got G set, you have to use gun set followed by the same name. You have to unpack it, and then you have to use less command. Once you have seen it, we'll quickly move on to spades. Okay. I'm typing spades. I'm checking if it is installed. I'm typing spades. You could also follow the same there. Okay. Spades give me an entire set of instructions on how to do this. Okay. Very key things we need to know here is that we are going to do a short read assembly. Okay, which is a de novo assembly. We have two pairs of reads for one genome, which is a mitochondrial genome, I think. Okay, so to indicate that, we have to give to show the forward pair, we have to indicate it to the assembler using -1. The next one with -2. Okay, so I'll go back again to where the cursor is. Now I'll type. I'll better see what are the files present in it. I have both. No, I have not done it both. Okay, fine. We'll complete the same. So this was spades. I'm indicating read pair 1 is short reads _1 dot first q. Read pair 2 is short reads 2 dot first q dot is that one is compressed, one is not compressed. I'm just trying if this works. Okay, and we have to give an output directory. Output directory we could give home followed by what you see here. For me, it is Gomathanaha because that's my name. I'll give the same here. Assemble. I'll give this name like this. Okay. Assemble. This is the output directory I'm going to give. I'll try running this. It runs. Okay. It has started to run. So, do you people have any problem with that? You could try the same. I'm typing the same command here. Spades. I'm indicating a pair, first pair of it. Short underscore reads underscore 1 dot first q dot g set. Okay. And for the second pair, I'm giving -2 short underscore reads underscore 2 dot g set. This is how it should appear for you. And I'm going to give an output directory home followed by your name, which is your name on the page, followed by assembly. Okay, I'm just giving you the command I just used, so hopefully you are able to follow it. I think so. You should see the screen running like this. Just like transforming, it is doing a K-mer index like I told you. Do you see that? And it uses a De Bruijn method of no such error directly being shown. Could you type ls and get me the output here to the chat box? Alexa, type ls where you are and get me the output here. So everyone else following me? So let us run. We'll quickly move to the presentation or some other things which I wanted to show you. If you find command line interface this way, stop sharing and I'll start sharing my browser window right now. Okay, because I want to show you some more things. So you could go to usegalaxy.eu. There are many other org, something like that, many other. Okay, I prefer EU. Okay. After typing the space thing, short tree sequences showing to you. Okay, you use Control plus Z. You press these two keys together, it should exit immediately and you can go back. Okay, this is the graphic user interface. This is also an open source. You could use this. You could also look for the same spades tool here. So you find a number of spades here. So it has a number of questions. Those are parameters you define, and you need to upload the data here using history. If you upload it here, it would appear in the history. So you could input those data here inside it. If you have got pattern format. Yes, you're Shivani, you are making error in typing the command. Spades followed by hyphen. You just copy paste the same command which I'm doing here, okay? Which I did a bit ago. I'll copy paste this. In the place of your name on WSN, you have to give your name on WSN. Just don't copy paste the same thing there. It wouldn't work. You could say no for directory found. Okay. Yeah. So that way we are looking into these things. We see PacBio reads. If you have got PacBio reads, you can give it as input. Nanopore, Sanger, it is for different things. And I told you what is a contig is. I did not tell you what is contig is. Contig is an assembled fragment. You get a raw read. In the short read cases, it will be a short fragment. In the long read, the fragments will be a little more longer. Okay. The contig is an assembled thing. Like we had trans-esterification, which is broken down. If it is broken into trans, ester, and esterification, these are known as contigs. So that is how an assembler normally would give if the data is not adequate to give a complete genome. Okay, that is what a contig means. So we'll look into it later. So I think you all have downloaded this genomics repository using your browser also. So you can go into this file. You must be able to see this pam.context.fasta. You could separately download it to your computer. And I'm just going to show you how to annotate it using dfast. This is a DDBJ maintained maintained server. So where you can annotate it. So once this for me page got completed. So do you have any response? Did the same happen? Could you follow? Were you able to follow it? So could you confirm it by telling a quick yes? Oh yes, great. Yes. So you mean the page has completed for you also after running this much. Could you confirm once again? Page finished running for you people. Okay, great then. So we have got our data in it. So what I just told you what a context means. Okay, we have got some FASTA files here. Okay, I'm going to open it for me. I have just opened it. I'll use less command. You just saw how raw reads look. Right after assembly, if you use less commands followed by that pathway, this is how an assembled file should look. So this is how a genome, this is a mitochondrial genome, I suppose, I think. So this is how a genome would look inside a mitochondria, means a sequence would look. Okay. Now later we could annotate this by supplying using several other tools. There are open source tools available. Even you can submit it to NCBI. NCBI could give it back. My screen is still on the first. I'm sorry. So now you're able to see. Okay, so thank you. So this is how an assembled file would look. I'm stopping it here. So you also see the same screen. If you see the same screen, just confirm by telling a loud S, a capital S. Everybody got it there? Everybody just assemble and we're able to visualize it. I'm just repeating what to do. In my case, this is a command I use. Yeah, I type less followed by the same command back again. I'll copy it and put that in the chat box. Yeah, you just saw how a raw reads looked. Okay, this in the same sample data. We'll have a look at it again. Less visualize short reads using short. I'll press tab. I'll say skill. Okay, so this is how raw reads look. Now this has been assembled right now into this. Less home gomutinayam. That gomutinayam space you have to give your name. How you have given out WSR followed by backslash assembly backslash context.faster. So from that flow rate, it has been assembled like this. And you could also see that a FASTQ file would start with at the right, a FASTA file would start with the greater than symbol, followed by the header. It will not bear any quality score. It will only have a plain sequence data on it, because this is a FASTA file, not a FASTQ file. So were you all able to follow? Could you tell a quick guess, or should I repeat it once more? Everyone? You want me to repeat it from where? From assembler. So could you spot the assembly file, context.fasta? Did you find where context.fasta is? No, you did not spot the context.fasta yet. So for the people who missed, means who couldn't find the context.fasta file, you type cd space tilde. Okay, and followed by that in the next line you type ls listed, and you just copy paste the output here. I want to see, and based on that I'll tell you. So at the same time, parallelly we will look into Saket. I think we have got a binary folder installed in it. I think you should type means maybe it is in a folder like you have to go to space followed by version. Move into the binary folder. Okay, in that binary folder there should be that you can execute page from space .py. You should find this is for Saket. You should find a space.py file. From space.py file, you have to do this thing you have posted. Okay, this is from Uganda has listed the output from, I think, a genomic sample data folder, I think. Okay, so you're stuck here. You were not able to do it. Is it so? Permission denied. Okay. Okay, fine, no problem. Pritesh, before you do that, you type PWD from the current working directory. Okay. You just that should return an output. You copy paste that output after this -o. Okay. So this error should get cleared. Okay. It is not able to make because you have got @ in between. Trends or something like that. After that name, you have got something which shouldn't be there. That's why that error has happened. Okay, we'll begin with annotating. I'll switch to the browser window. So I just had downloaded a sample what. Okay, it is human. Okay. So I just have got the file. I'm going to the first page. So you're able to see start your project thing. Okay. You can't do what? Sorry. Could you elaborate? I'm going to start your project. Okay. I'm going to choose the file. I just had downloaded that par.context file from my genomics repository. I go to desktop. I got this FASTA file, context.fasta. It should be par.context in your case. Okay. I'm giving DNA name. You can give your mail ID. You can select platform taxonomy check also with completeness check. You could run it. So your data would get annotated and it would simply be available on this web page. So I think I've touched upon most of the things. Maybe few of you might be having some errors here and there. The same thing you could do here also. There's for prokaryotic annotation, there's a tool called Procar in Galaxy server. We have got Procar here. You could use the Procar for same annotation. So I touched upon how to deal with the raw reads, how a sequencer gives the raw reads, what to do with the raw reads, how to process it, and once you have get the raw reads, how to assemble it, and an example way of annotating it. There are a few other things I could share, but I don't think time would permit. There is this company called Vellore. This is a subsidiary of Google. They're doing with something like this, and you might have heard Ms. Meena discuss yesterday. She was working with this kidware. They also have this precision medicine thing. You could work with them. There is Google Cloud Life Sciences. The same package will be available there as well. Same thing in a different thing, a different platform. In a Google platform, you will find it. Okay, you could use this to analyze your clinical genomic data to find the variance between that. Similar, much more things you could do. This is where they are also working with, they also have got some tools like that. This is very lab sensors. They have got tools for this particular precision medicines. So there's a lot of business opportunity, like both of the speakers told from yesterday and today. I hope this was quite a useful thing for you. I hope the same. Maybe you still have got some doubts. I think you could sincerely post it on my genomics repository under issues column. I could do that. So thank you very much. I think I've just given you an introduction on what are all things to be done with Linux environment and what are the basic tools available. Also for variant calling methods, which these days these big guys are working with, there's something called a variant calling function. So this is something like finding a mutation between a normal and cancerous thing. There's a tool called Deep Variant. Deep Variant is developed by Google. You could use that. There's a tool which is used to find this. It's Google's repository in GitHub, like how I showed you there. So there were also issues. They have posted there. So this is a deep variant tool to find variant calling, like how they told Dr. Nalasampanasamy told to find the difference between mutated genes, like that. Similar things are done using this deep variant. So I think there's one more query from Yogananda. After assembly is done, I just wanted you to visualize it using less command. Okay. So after that, inside this repository which I had showed you, you could download this repository data. Okay. There's a raw data. You could download it to your local computer, and which would be in the name pan.context@FASTA. This is a sample assembled bacterial genome done at a laboratory. Okay, I just updated, means I just updated it to DFAST. You could, it's for your practice. You could upload it there and DFAST and get it annotated for your information. Okay, you could do the similar thing for your. If you find coronavirus reads also, you could try assembling using spades. You just go to NCBI. I don't think this novel coronavirus is available with sequence reads right now. You go to SRA. SRA stands for Sequence Read Archives. You go to this. You could try any viruses. Virus could be simple for your WSL command. It's the WSL environment. Okay. You will get a number of, you have got here raw reads from a nanopore sequencer of size 33 MB. Okay. You could download this and you could use same spades to assemble it. And there are some other tools to do it, like there's a pipeline called Unicycler. I use it normally. Okay. Also, you could use the same tool. You could just type sudo apt install Unicycler in your terminal to use it. Okay. This is also another pipeline to specifically the assemble read from Minion. Okay, this is the site where you have to look for raw reads. Okay, I think I have tested on most of the things already this time, so I better end here. Thank you very much. If you have got queries, please post there in issues. Okay, thank you very much.
Yeah, thank you, Gomti Naigam. Am I audible?
Yes, I'm audible. Yes.
Hello. Am I? Okay. Okay. So thank you very much. Really showing, giving us the comprehensive application of the NGS and showing us step by step the NGS data analysis. I'm sure it will be very helpful for all the participants. And of course, you need some practice. I don't think that in one go you will be able to master and follow this. But after few practice, I think you will be able to follow and carry out your projects in a much better
Yes,
way.
sir. Yes, exactly.
So really, thank you very much, Dr. Gomti Naigam. Can you take some questions if there are some questions?
Yes, ma'am,
So
sure. I think
can
they've got some
students,
issues. Yeah, thank you.
yes, yeah. So Tamba Medha, will you ask the questions, read out the questions?
Sakshi, can you help, ma'am?
Yes,
Can you
yes.
take
Yeah,
the
I was
question?
just looking into it, ma'am. There are not, they had doubts
Okay,
regarding it, ma'am, but there are error codes in itself, not exactly questions. Which are
They've got some issues. So
Yes, yes,
for
yeah.
that cases, they could go here and
yes.
put it under issues. So I could solve it later on.
Okay, okay, sir. I'll just do that.
So how to, there's one question asking
Yes.
how to analyze RNA sequence data. It depends on
Yes.
what you want to do. If you want to do annotation from that, there's a different setup too. If you want to do, there's a tool called Trinity to assemble RNA data, Trinity GitHub. You go there, or else if you're not, I think Saket is familiar with the Linux environments. If for people who are not familiar with that, you could go to RNA Galaxy website, RNA-seq. Yeah, you will find lots of tools to analyze RNA sequence data. If you want to know the differential sequence, there is setup tool. DBSEQ, there's a tool called DBSEQ, and if you want to just annotate it, there's this Trinity RNA-seq. You can assemble the sequence, de novo assembly of transcription data, assemble it and annotate it, or also you can use it if you want to use for annotation cases. There is a tool called Raker GitHub. So you could use this. So these are all the tools available for RNA sequence. And also I wanted to tell about metagenomic sequence processing. You could use this KIME workflow. There's this tool. KIME is also available in Amazon Web Services as a cloud platform. KIME AWS, you could go there. And also this NGDA server. I wanted to show you this data, but we ran out of time. We have some analysis there in KIME and NGDA as well. Show you. If you have got some metagenomic data, you could use this NGRAST and also KIME. Go straight away to analysis. The website is quite slow. The server is down, I think. Seems the server is down right now. You could follow it later also. Okay. So any more questions?
No, sir, except that I don't see a question.
Yeah, even if they have got any, they could post it
Yes,
on
sir.
issues, I would say.
Yes, sir. Sure, sir.
So I just wanted to make sure from them that they have learned a bit at least from processing the array to assemble it and using annotation tools. So I'm very happy of that. Thank you very much. And I take this opportunity to thank Dr. Radha again and all the panel members. Ms. Vaishnavi, Ms. Sakshigara, and Veda for making it very smooth. And also thank Professor Ma'am for leading the session. Thank you very much once again.
Thank you, sir.
Okay, so I thank, finally, I thank Mr. Gomti Naigam for his sincere effort and the really enlightening workshop. And I hope that is very, very useful for all the participants. And also I thank all the participants to join this workshop. Thank you very much.
Radha ma'am, if you would like to share a few words for Gomti sir.
Sure, sure. I was waiting for you. Thank you, Gomati Nagan. Great. I think I have to come to TVA to learn. Lot of knowledge, lot of experience. I'm so proud of you. Thank you so much for your time and your support in coming up with this. Thanks a lot. And thank you, Madam Alka ma'am. I'm sorry we could not see you on your video, which we would have loved to. But thank you, ma'am. And thanks for your time too, ma'am. And hope the participants really enjoyed this workshop. And I think Gomati Nagan, you'll be having many more questions. Probably we will send that to you, your mail, right? Sure. Thanks a lot. And thank you, students. If you have any anything to announce.
I'll just take a moment, ma'am. So coming to the end of the workshop event, I would just like to take a moment to appreciate all the participants for their attention, their questions, and their engrossment. Thank you so much, sir, for sharing your knowledge and expertise in the field. We are very thankful for the precious time you could devote for all us learners. I'm sure all the participants must have enjoyed themselves in working their way in it. We hope to have such sessions in the coming times as well. And a very special thanks to Alka Mehta ma'am for helping us coordinate today's workshop, and event organizer Radha Saraswati ma'am as well. Once again, thank you everyone. Stay home, stay safe.
Same. Thank you once again.
So before going to the further events, we'll have a small break to energize and relish ourselves, and we'll meet you back at 1:45 p.m. onwards for the keynote lecture 2. Thank you so much, everyone. A small announcement for the participants. There is a feedback form posted in the chat box. If you would just take a moment and fill that up for your attendance, please.